topics preserved. Sorry, there are several topics reserved for today's meeting. Okay, the first thing which I'm going to tell you about is just the topic of some molecular anthropology studies related to forensic genetics. The second thing is we are going to continue with the exercise with the exercises that we've started last time, and we have also some presentation of interesting cases from the literature, from our casework, just to show you what may happen, how to deal with the difficult cases, and so on. Okay, so let's start with the first topic. Have you thought about the term of the exam? I will send you the link where you can check. Okay, this will be a link because okay, this is the link where you can check the terms when the lecture room is free. Okay, actually it is occupied practically every day till 4 p.m. Okay, so probably just look at the lecture room, sala wykładowa, and just so and then please check which term is convenient for you. I hope one of the afternoons it will be suitable for all of you, so that you can come to the Department of Forensic Medicine, so that we will have the exam, of course, after the last seminar. Okay, so have you thought about any terms or not yet? Pardon, I cannot hear you. Could you please repeat? I cannot hear you.
We need more time. We didn't think about it yet.
Okay. I can hear you now. Thank you very much. Okay. So at this website you can check the availability of different rooms at the Department of Forensic Medicine. Okay, and the room which is the best for us will be the lecture room, sala wykładowa, because there is a lot of space. Okay. And you can have this distance between, you can keep the distance between students. Okay, so let's start with the first topic for today. Okay, so as I promised, we are going to talk a bit about. Sorry. We are going to talk a bit about molecular anthropology, and just the topic which I will focus the most will be DNA typing for resolving geographic origin of distant ancestors. Okay, so just to remind you what DNA polymorphism is. Okay, by definition, it is occurrence of two or more alleles of a particular locus in a population with frequency higher than the one resulting from general mutation rate. Because very often it's quite difficult to us to assess what frequency results from is higher than resulting from the mutation rate. It is assumed that DNA polymorphism means occurrence of the most common allele with frequency lower than 99%. Okay, so everything which is which has frequency higher than 1% is considered polymorphism. Okay, something which is lower, just by definition, is considered as a mutation. Okay, and 0.3%, okay, of the human genome shows polymorphism, which corresponds to approximately 1 million nucleotides in the whole human genome. Okay, and of course we have two types of polymorphism. We have sequence polymorphism, which is studied in the especially in the mitochondrial DNA. Okay, in forensic genetics, and the length polymorphism, which is studied by forensic geneticists when in case of nuclear DNA. Okay, and of course you remember that the short tandem repeats, which are commonly used in forensic genetics, are the markers which show length polymorphisms are divided, which are used and analyzed in forensic genetics are divided into mini satellites and microsatellites, and of course microsatellites are more are generally the markers which are markers of choice because of the length of the repetitive unit, which is shorter, much shorter than in case of many satellites, so that the size of the alleles of the PCR products are lower. Yeah, the sizes of the products of the fragments amplified by PCR are shorter, so that in case of the degraded DNA, there is much higher success rate in case of analysis of microsatellites, also commonly referred to as short tandem repeats STR markers. Okay, so let us go further. So this is how we just remind you how microsatellites look like. Okay, and they are called, of course, the alleles are designated based on the number of repetitive units. If the number is, if there's, for example, here's an example. If we have 7 repeats, it's allele 7. If we have 8 repeats, it's allele 8. Okay, and of course we have different number of repeats on different. There can be different number of repeats on different chromosomes and also in different individuals. Okay. And of course, you remember that typically STR markers are analyzed in forensic genetics by multiplex PCR. They are amplified. Many STR markers are amplified simultaneously in one reaction. Okay. And typically, STR markers are analyzed by in a way that we have that one of the primers used for amplification of an STR marker has a fluorescent dye attached to the 5' end of the primer, so that the resulting PCR product is also labeled. One of the single strands is labeled at 5' end with the fluorescent dye. Okay. And of course, you remember, this is just a brief summary, which is what I guess you know, that the resulting PCR products are separated typically in forensic genetics by capillary electrophoresis, and of course, the shorter fragments go more quickly through the capillary, so that they reach a place called window, on which argon laser shines and induces fluorescence, which is detected by the detection system, and it's stored on the computer. Okay, and here is an example of electrophoregraph of PCR products labeled with 4 different fluorescent dyes. So here you can see the pics. You will see such results during the exercise because you are going to solve one of the cases which I will give you. Okay, so this is just here you can see just 4 markers which are separate, which you can distinguish which is which, because they have different allele size ranges. Okay, so the shortest fragments, this is the size in base pairs. The shorter fragments. Carolina Milch also came. Okay, I will mark because I can forget it. Okay, sorry. Okay, Carolina Milch. Okay, we have you. Okay, so we can distinguish one from another by the size. Okay, of the shorter products belong to the marker D8. Okay, the longest products belong to the marker CSF1PO. Okay, here you can see heterozygous situation. Yeah, At this locus also two alleles, heterozygous individual, and here we can see a homozygote at this locus and homozygote at this locus, because we amplified only one allele. Okay, so we have in total 4 different dyes were used for labeling PCR products. Okay, so we have and the 5th dye is reserved for a size standard. Okay, so we mix our PCR products with a size standard, which is labeled with another fluorescent dye, so we can know, so that we know the sizes, exact sizes of the products, which of the PCR products which we amplified during our multiplex PCR. Okay, let us go further. And of course, the alleles are labeled. The alleles are called by comparison to an allelic ladder, which is a mixture of alleles found in the population. They are sequenced, and so we know how many repetitive units are present in each allele. Okay, so for example, this is allele 14, so we know that there are 14 repetitions of a unit. Okay, and by comparison of our allelic ladder, okay, and the sizes of the size of the fragment with the size of the fragment amplified during the amplification of our sample of interest, okay, we can see, oh, the size is more or less the same. Okay, the gray field is called bin. So okay, oh, this allele, the peak of this allele is within the bin. So the computer software calls this allele 14. Okay, and here you can see an an example of an off-ladder allele, which falls in between different alleles found included in the allelic ladder. Okay, so I guess this allele should be called 10.2. Okay, because it is here you can see a bin for the allele 10 plus 2 more nucleotides, because it will be longer. Okay, this is the size. When 55 nucleotides, we can see here 257 nucleotides. Okay, so this allele should be called 10.2, which I guess you remember, 2 indicates 2 additional nucleotides. Okay, let us go further. Okay, so how do we uncover history of the humankind? Okay. One of the sources, of course, is the oral lore. Okay, so we can talk. For example, you can talk to your parents, to your grandparents, and they will tell you some history, okay, of their lives, okay, and what was happening in the country and so on. I guess they remember a lot. Okay, and of course, but you will not know very often what was going on earlier. Okay, so another way how to uncover the history is that the history is all very often preserved in the written sources. Okay, so that some people write the history. Okay, so so that we can reconstruct the history. And another source, of course, is archaeology. We can dig and found some artifacts, some bones, and also reconstruct some historical events. Okay, and some facts about history of different, about especially contacts between different human groups is uncovered thanks to linguistic studies. Okay, by comparison, oh, this word in the Polish language, for example, there are some words in the Polish language that came from some Iranian languages. Oh, probably Slavic, Proto-Slavs must have lived nearby some Iranian peoples. Okay, or some. So this is how it's reconstructed and studied. Okay, and another source of history is quite recently is DNA. Okay, and how the genetic markers are inherited from ancestors. Of course, you know that generally different markers are inherited independently based on the law of independent assortment, which is the second Mendel's law. Okay, so generally you can say, oh, different traits, different markers are inherited independently. Okay, but of course, you know, it is not always true because of different factors. Okay, and the other fact which you are aware of is that the recombinational variation resulting from crossing over during meiosis, which is which was described in the chromosomal theory of inheritance in 1915 by Thomas Morgan. Okay, and based on these two laws, okay, these two theories, we know that DNA from of our ancestors is mixed. Okay, we are a mixture of DNA of different our ancestors. Okay, so actually I do not know which part of my genome came from my okay, which part of the genome came from which grandfather or grandmother, okay, or great grandmother, great grandfather, because we have, of course, you know, we have two parents, we have four grandparents, we have 8 great grandparents, and so on. And actually, it's quite difficult to assess which part of my genome came from where, from whom, okay. And of course, there are exceptions. Okay, so which you already know and you are aware of. With exceptions, these are fragments of our genome which lack recombination. And they form haplotypes which are transferred intact to next generations. And of course, this is the Y chromosome, which is inherited paternally. It's transferred intact only by males. And we have also mitochondrial DNA, which is transferred intact only by females. And generally, these two fragments of our genome are commonly referred to in molecular anthropology as lineage markers. Okay, and just to sum up, you know that males inherit Y chromosomes and mitochondrial DNA, but they do not transfer mitochondrial DNA to next generations, and females inherit mitochondrial DNA and of course transfer it to next generation. Okay, and just to I will skip it, okay? And haplogroup, a word which I will use quite often, okay? Haplogroup, by definition, is a group of haplotypes. For example, mitochondrial DNA haplotypes or Y chromosomal haplotypes with an identical set of alleles inherited from a common ancestor, okay? So, for example, if my Y chromosome, maybe the Y chromosome of Casper, okay, this is just the first person which I can see on the list, are the same, they have the same set of values at different positions. It means that me and Casper had the common ancestor, okay? So we belong to the same haplogroup, okay? So the haplogroup is the same, so it means that we come from the same paternal ancestor, okay? This is just the genetic structure of the mitochondrial DNA, just the first mitochondrial DNA molecule which was studied, it was sequenced, consists of 16,569 base pairs. Of course, we have 22 genes encoding tRNA, 2 genes encoding rRNA, and 13 genes encoding proteins. Okay, and you remember that in forensic geneticists are interested especially in the control region. We have hypervariable regions 1 and 2. Also, there is another hypervariable region 3, which is analyzed by forensic geneticists more rarely, but it's sometimes analyzed. Okay, so this is a fragment which does not encode any genes. This is this control region in the deal of the mitochondrial DNA. Okay, so how do we identify the mitochondrial DNA hub? Okay, initially it was done in this way that the mitochondrial DNA or products of amplification of mitochondrial DNA were digested with different restriction enzymes. Okay, so this was this restriction fragment length polymorphic markers. Okay, and depending on whether the fragment was digested or not, the mitochondrial DNA was digested or not, different haplogroups was detected, and the person and this person's mitochondrial DNA was assigned to. Okay, and also another way how to identify the mitochondrial DNA to sequence the control region, the hypervariable segments HV1 and HV2, which are analyzed routinely in forensic genetics. Okay, and of course you can do it directly by sequencing, for example, the whole mitochondrial, the complete mitochondrial DNA genome, so that you know exactly, you know all the positions, all the molecules, and you can know know exactly which mitochondrial DNA haplogroup this molecule belongs to. Okay, and the second way is actually indirect way of prediction of the mitochondrial DNA haplogroup, because this sequence of hypervariable region 1 or hypervariable region 2 does not indicate us directly the haplogroup. This is only prediction. We simply predict, oh, this sequence is typical, for example, for haplogroup U or something like this. Okay, let us go further. Okay, so you can see here the control region of the mitochondrial DNA with the location of HV1, HV2, and HV3. The segments which are routinely analyzed by forensic geneticists. Okay, and they can be sequenced with the use of Sanger sequencing, which I'm sure you know how it works. Okay, and by studying mitochondrial DNA in Europe, researchers, okay, let's start with the fact that in 2001, a geneticist, Brian Seitz, published a popular scientific book entitled The Seven Daughters of Eve, okay, in which he actually described like you can say, a fact that the vast majority of Europeans, modern Europeans, can be traced back to only 7 different females, 7 different women. Okay, and he gave different names to these females. Okay, Ksenia, Ursula, Helena, Tara, Yasmin, Katrin, and Velda. Okay, so this is just, and depending on, and actually based on the variation of the hypervariable segments 1, 2, and 3 within these haplogroups, we can predict. Actually, we can predict when and where this haplogroup occurred for the first time. Okay, and later studies, of course, found more haplogroups. Okay, and for example, in a later study which was published 3 years later, actually found that there are 10 main haplogroups present in Europe. Okay, in Poland, more than 70% of people originate from 3 females, and the one which is the most common in Poland is the haplogroup H. This is this Helena, which actually, according to molecular anthropological studies, lived 20,000 years ago, somewhere in France. Okay, so this is how and so based on so the geographic origin and age of a haplogroup are determined by analysis of polymorphism of the control region in various geographic areas. Okay, so based on the so-called molecular clock, you know how frequently mutations occur. Okay, so especially of the mitochondrial genome. Okay, so you know how frequently the mutations occur. So you can reconstruct, you can go back and calculate the time where this haplogroup occurred for the first time. Okay, appeared for the first time, when this mutation must have occurred. Okay, and you can also, and based on variation of this haplogroup, of the variation of the control region within this haplogroup in different regions of the world, you can predict the geographic origin of the haplogroup. Okay, the more diverse this haplogroup is in this region, okay, it is the more likely this is the homeland of this haplogroup. Okay, let us go further. So you can see a phylogenetic tree of mitochondrial DNA haplogroup. Okay, of course, some of them are found, for example, only in Africa, sub-Saharan Africa, some in Eurasia, and so on. Depending on of course, this mitochondrial DNA show high level of geographic specificity. Okay, and this mitochondrial DNA haplogroup studies were, for example, found they were applied in the studies of origin of modern humans, and actually this mitochondrial DNA studies provided evidence for the out of Africa model of monogenesis of all modern humans, and the falsity of polycentric hypothesis. Okay, because there are two hypotheses which were formulated by different anthropologists. One of the theories says that modern humans developed in one place in the world. Okay, and then the modern humans spread throughout the world. And the other theory says that the modern humans developed independently in several places of the world. And actually, mitochondrial DNA studies show that all modern humans can be traced back to one genetic Eva who lived many years ago somewhere in Africa. Okay, so this is what this is just the main conclusion. And of course, the second lineage mark, which I am going to tell you a bit, is the Y chromosome, which is actually one of the smallest human chromosomes in our genome. Only chromosomes 22 and 21 and 22 are shorter than the Y chromosome. And as much as 95% of the Y chromosome, of the human Y chromosome, does not undergo recombination during meiosis. So actually, it is the largest haplotype in the whole human genome, which is more than 3,000 times larger than the mitochondrial DNA. Okay, and of course, we have different polymorphic markers present on the Y chromosome. The human Y chromosome, we have sequence changes, the SNPs, single nucleotide polymorphism. We have indels, insertion deletion polymorphisms. Of course, we have also microsatellites studied in forensic genetics, especially, and also we have mini satellites. Okay, and I think I have told you about this database. Let us go further. Okay, you know that the Y chromosome, I told you this, sorry. So I just will remind you very briefly that the Y chromosome contains markers which are specific only to males. So this can be used in forensic case, especially when we have mixture of DNA of a man and a woman. For example, in rape cases, especially, okay, that when the perpetrator is a male and a victim is a female. And we have also the Y chromosome shows a unique inheritance pattern because it's transferred paternally, which can be used in paternity testing when the putative father is not available for examination. Instead of him, we can test his brother, his father, his grandmother, his paternal uncle or paternal cousin, or another son if he has another son, just when the child supposed to be the child of this man is a boy. Okay, and also the Y chromosomal haplogroup studies may be used are used. The studies of the Y chromosomal haplogroups are used to discover the prehistoric migration patterns of modern humans, and also they provide the additional evidence for the out of Africa model that all modern humans can be traced back to genetic Adam who lived many years ago in Africa. Okay. And what is important and special about the Y chromosome is that the Y chromosome may show significant differences even between closely related populations. Okay, the first study published in 1996 compared Dutch and Germans. The Dutch and the Germans, and they found that actually Dutch and German languages are quite similar. Okay, they are both Germanic languages and they are neighbors. Yes, so everyone would think, oh, it is likely that they are genetically similar. But this study from 1996 found that these populations differed in the Y chromosome, which was really quite surprising. Why? Wow, these populations are historically and linguistically related so much, okay, but they differ in the Y chromosome, okay? So it makes this Y chromosome a very useful tool for genetic studies of relatively recent historical events, okay? And here you can see results of a study from 2001, comparing the YSTR loss differences between different European populations, not only European, sorry, different human populations, and based on variation of 7 Y chromosomal STR loss. Okay, so you can see, for example, here we have Dutch, Switzerland, Italy, Germany, and so these are some Europe. Okay, let us go further, because just so why do humans show so many differences, interpopulation differences in the Y chromosome, but when you look at the mitochondrial DNA, these populations do not differ so much. Okay, so actually, the anthropologists suggest that this results from the fact that women were traveling much longer distances than men, okay, during the history of our species, okay, and this is strongly supported by genetic studies, okay. Genetic studies demonstrate a higher female than male migration rate, and it is explained by historical patriarchality, a social system in which a married couple resides with or near the husband's parents. Okay, in the history, it was always a woman who was traveling to her future husband's place. Okay, so actually, this patriarchality is, I think, it's still preserved in some human cultures. Okay, for example, in India, yeah, I know that, for example, it's like it impacts a lot, just like the social, how to say, treatment of different sexes in India, okay, because it's quite for example, married couple very often abort female fetus because they know that their daughter will be transferred to another family, and they will have to give they will have to spend a lot of money to educate this woman, yeah, and so on. But actually, for the family, it will be a loss of money because she will go to another family afterwards. So this is just actually a sad result of the system that, for example, the higher abortion rate of female than male fetuses only based on the sex, only because of the gender of the fetus. Okay, so this patriarchality actually is in Europe is something historical. And it's not practiced, I guess, in majority of the local groups in Europe, okay, and vast majority. And just to tell you that, for example, this patriarchality is preserved linguistically in Slavic languages in the verbs of marry, for marry, okay. For example, for males, we have in Slavic languages, just like the one in like Polish language or the Russian language, Czech, and many other languages, we have different verbs for marrying, for a man and a woman, okay? Because we say that the man żeni się, okay? It means that I would translate that he becomes wife, something like this, yeah? That he becomes wife. But for the same about Russian, okay? Żenicja, okay? No, the translation, to wife oneself, okay? And for females, we say that the woman wychodzi za mąż, она wychodzić za mąż in Russian, okay? It means actually that she quits and follows her husband, okay? So this patriarchality which is not practiced anymore in Poland. Yeah, it's preserved in the language. Okay, that the daughter, the woman was leaving the place that she was living, where she was living, and she was following her husband. Yeah, so just it's also preserved in some songs, traditional songs, wedding songs. Yeah, that the woman that is getting married just very often cries and say goodbye my mother, goodbye my brother, goodbye my father, oh I'm leaving you, and so on. Okay, and the man say, oh stop crying, and we are going to our new home. Okay, and how do we identify the Y chromosomal haplogroup? Of course, the most we can do it directly based on the analysis of event unique polymorphisms. Okay, unique event polymorphisms. These are polymorphisms which are assumed to appear only once in the genetic history of our species. Okay, and these are mainly Y SNP loci with mutation rate of approximately 10 to the power of -8. However, some recurrent mutations are known, but they are rare, but recurrent, that which reversed actually the mutation back to the original state. Okay, and another unique event polymorphism which is studied and it's located on the Y chromosome is the insertion of the alu sequence, which defines so-called haplogroup DE. Okay, haplogroup DE of the Y chromosome is defined by the insertion of alu fragment on the Y chromosome, and of course we can also determine the haplogroup just in case, similarly as in case of mitochondrial DNA. In case of mitochondrial DNA, we can predict the mitochondrial DNA haplogroup based on the variation of the control region, the hypervariable segments, which are analyzed routinely in forensic genetics. And also in case of Y chromosome, we can predict the haplogroup based on the variation of STR markers, which are analyzed routinely in forensic genetics. Okay, and these markers are assumed to have a mutation rate of approximately 10 to the power of -8. Okay, and so we can predict the haplogroup based on differences in allele frequencies in different haplogroups. Okay, here is this is a very basic phylogenetic tree of Y chromosomal haplogroups. Okay, haplogroups A and B are found natively only in sub-Saharan Africa. Okay, for example, haplogroup C is found especially in Eastern Asia. Okay, haplogroup E is found in Africa, but also in Eurasia. Haplogroup H is found in Southern Asia and also in Europe, especially in Gypsies, in the Romani people. Okay, haplogroup I is found in Europe. J is found, for example, in high frequencies in Semitic populations, but also in other populations. N is found in Northern Eurasia. Q is typical for Native Americans. Okay, and haplogroup R are found also in haplogroup R is found in Eurasia. Okay, but this is just very basic tree. Okay, because we can divide these haplogroups further by detecting more sub-haplogroups, more subclades. Yeah, because they are still, because every haplogroup still have some descendants with different mutations and so on, and these mutations were also inherited by the new descendants, which was basis of forming of another haplogroup. Okay, and here you can see a tree, Y chromosomal phylogenetic tree published in 2005. Yeah, you can see how many haplogroups were known in 2005. And right now, we have really very many haplogroups. Yeah, I can just make a zoom on one of the branches of the haplogroup. You can see just this is the, but it is still quite old tree, very old tree, because right now there are very many haplogroups and sub-haplogroups and subclades of the Y chromosome known. Okay, so how can we identify the Y chromosomal haplogroups based on the analysis of YSTL loci? Okay, you know that we have different just look, please take a look. This is one of the STR marker which is used in forensic genetics, DYS 437. Okay, and here you can see that in different haplogroups, there are two haplogroups, haplogroup G and haplogroup I1C. They are totally different frequencies of the of different alleles. Okay, so if I detect haplogroup, if I detect allele 14 in my sample, it is much more likely that this person belongs to haplogroup I1C than to haplogroup G, because in haplogroup G this allele occurs, but it's very, very rare. Okay. Similarly, allele 16 indicates with much higher probability that this is haplogroup G than haplogroup I1C. Okay. And the same, for example, these are examples of European haplogroups E3b, I, R1a, and R1b. Okay. For example, if I analyzed marker DYS 426, which is a marker which mutates very rarely, okay, and actually majority of humans have allele 11 and 12, okay. If I detect allele 12, okay, it is much more likely that this is haplogroup R1a or R1b. It's actually very, very likely, very unlikely, extremely unlikely that this is haplogroup I or E3b, okay. So if I detected 12, so I'm sure that it must be R1a or R1b, okay. And then when I detect allele at another marker, which is also a marker which mutates quite rarely, more often than the previous marker, but still quite rarely. If I detect allele 13 or 11, yeah, I know it must be either R1a or R1b. Okay, so this is just, but of course, this estimation of probability of haplogroup assignment, correct haplogroup sequence, is based on statistics, on Bayes theorem. So it's not just based so of course proper computer software is used for assigning different YSTR haplotypes to different haplogroups. Okay, so of course, just to sum up, the Y chromosomes can be used in genealogy to uncover the deep ancestral origin concerning defined by thousands of years. Okay, so we can detect the haplogroup. Okay, so this is the ancestral origin based on the Y SNPs, and of course we can also use it to trace family genealogy. Okay, recent ancestry based on hundreds of years, and the YSTR markers are especially useful to reconstruct the recent YSTR, the recent paternal ancestry. Okay, and just to show you example how we were revealing, how we were detecting the haplogroups at our department. Okay, we used a multiplex system which was worked out at the Department of Forensic Medicine at our university for simultaneous analysis of 18 YSTR loci, and there were 8 YSTR loci with low mutation rate. Okay, these two markers, one of them which mutates extremely rarely, okay, DYS 388, DYS 426, and also additional 6 markers which mutate relatively rarely. Okay, so these markers, because they mutate so rarely, are especially useful, particularly useful for assigning YSTR haplogroups correctly to different haplogroups. Okay, so examples of methods used the most commonly in genotypic YSNP markers, the RFLP analysis. And here is an example from our practice. Okay, this is a mixture of 2 products which are possible to detect. Okay, this is a polyacrylamide gel, and DNA fragments were stained with silver. Okay, so if the product or PCR product is digested with the enzyme, the fragment is shorter. Okay, so you can see an individual which has ancestral state at this TAT marker. So it means that these individuals do not belong to haplogroup N3. Okay, and these 2 individuals, DNA of these 2 individuals was not digested, so the fragment is longer. So it means that these two haplogroups from Poland, these two individuals from Poland, yeah, belong to haplogroup N3, which is typical for Northern Eurasia. Okay, and of course we can use many sequencing to detect exactly the haplogroup, to analyze exactly the locus responsible for the haplogroup. Okay, and here is an example of a mini sequencing-based multiplex snapshot assay. Okay, many markers are amplified simultaneously in one reaction. Okay, and depending on the allele which is detected, this can be with the G, the color can be specific, the product can be specific for the G base pair, sorry, for the G nucleotide, A nucleotide, C nucleotide, or T nucleotide. Okay, we know what the exactly which haplogroup the sample belongs to. Okay, and of course when you analyze a single nucleotide polymorphism by PCR, the resulting products are very often very short. Okay, so it's suitable for an analysis of degraded DNA. However, when all the products were equally short, we would have it will be quite difficult to analyze these results, because we will have plenty of products with similar size. Okay, so this is why when we analyze these SNPs with many sequencing, some additional neutral sequences are added, which make the resulting fragments longer. Okay, so this is just like a neutral sequence which is added to make the resulting fragments short. Also, quantitative PCR with use of Tachman probes is a method used for detection of different single nucleotide polymorphisms, also to determine the Y chromosomal haplogroups. And here you can see actually a result for an autosomal marker that we have here: a homozygous individual with one allele, homozygous individuals with another allele, and here you can see heterozygous individuals, and here is non-template control. So these are samples without DNA. Okay, and of course, in case of Y chromosomal SNPs, this we will detect only this group and this group. There will be no individuals. There should be no individuals with the mixture of two alleles, because you know that the Y chromosome is only one in males, and in males, and so it's not possible to observe two alleles at once. Okay, and just a high throughput method, the Tachman open array, for simultaneous analysis of 16 up to 256 markers. Okay, and of course, currently the high throughput sequencing, next generation sequencing, is also used routinely for Y chromosomal Y chromosomal haplogroup assignment of a sample. Okay, when we look at the Y chromosomal haplogroups in Europe, a study from 2000 by Semino and colleagues demonstrated that more than 95% of males in Europe originate from 10 paternal ancestors. What do you think of making a short break? Because I talk a lot. Is it okay to have 5-minute breaks or 10-minute break? Okay.
Yes, it's okay.
Okay, so let's meet in 10 minutes. Welcome back. Are you here with me? Okay. I'm calling. Share screen. Okay, we are continuing with the Y chromosomal haplogroup. So the study from 2000 demonstrated that more than 95% of males in Europe can originate from 10 different paternal ancestors. Okay, so these are these haplogroups which are assigned different colors. Okay, and you can see that this distribution of these haplogroups in Europe can be totally different depending on the populations. Okay. Regarding the Y chromosomal haplogroups in Europe and generally the Y chromosomal studies, we can say that the modern humans, Homo sapiens sapiens, appeared for the first time in Europe approximately 50,000 years ago. Okay, so these were first modern humans, Homo sapiens sapiens. Okay, and during the last glacial maximum, which took in Europe, which took place approximately 18,000 years ago, human populations survived only in several places on the continent. Okay, so these are these populations labeled with different colors here, with the violet, red, and pink. Okay, and after the last glacial maximum, okay, after the was finished, okay, the Europe was recolonized again from small populations which survived this period. Okay, and it is assumed that this must have been the popular haplogroups R1b from the Iberian Peninsula, I from the Balkan Peninsula, okay, and also R1a from this area in present day at the Black Sea. Okay. So then afterwards, there were subsequent migrations to Europe, haplogroups E3, BF, J2, G2. These were people from the Middle East, actually first farmers in Europe. So they arrived to Europe approximately 8,000 years ago. Okay, so they arrived, so they brought farming with them. Okay, actually I checked my Y chromosomal haplogroup, and my Y chromosomal haplogroup is this Middle Eastern haplogroup. So actually my paternal grandfather was a farmer. My father was the first one who stopped this tradition. Yeah, so actually I can guess, yeah, maybe it was like this, that since the arrival of the first farmers in Europe, my ancestors were the farmers until my grandfather. Okay, and then approximately 4,500 years ago, people from the Euro region arrived to Europe. This is the haplogroup N3, and which is assigned to this Finno-Ugric speakers. Okay, Finno-Ugric languages are languages such as, which include, it's a family of languages which includes Finnish, Estonian, and also Hungarian. Okay, let us go further, and here you can see the distribution of frequency distribution of haplogroups in Poland. Okay, as the most common haplogroup in Poland is R1a. Approximately 60% of males in Poland belong to this haplogroup. Okay, you remember this is a haplogroup which is assumed to have survived the last glacial maximum. Okay, then we have also haplogroup I, but different studies actually question it, yeah, whether it was in fact like this. Then we have also haplogroup I. Okay, right now I can tell you about this haplogroup. This is the one which is assumed to have survived in the Balkan Peninsula, okay, during the last glacial maximum. And different studies actually support the theory that this is for sure, this is the only haplogroup in Europe which is a pre-Neolithic haplogroup. This was the first Europeans which were mainly hunters and gatherers. Okay, so before the introduction of farming to Europe. Okay, so this is this Paleolithic haplogroup I. Okay, then we have also haplogroup R1b, the one typical for Western Europe. Yet 12% of males in Poland belong to haplogroup R1b. Then 9% of males, these are this farming-related Middle Eastern haplogroups E, G, and J. And then 4% of males in Poland belong to this Uralic haplogroup N3. Okay, so I will tell you just, okay, so when we detect that the person belongs to haplogroup R1a, what can we tell to this person about the history of this person, about the origin of this, about the genetic origin of this person? Okay, so generally haplogroup A was defined by marker M17. There is a stretch of 4 guanosines in one of the loci on the Y chromosome. And when there is deletion of one of these guanosines at this site, this is actually which occurred only once in the history of our species, okay? In the genetic history of our species, this is just a genetic marker of haplogroup R1a, okay? And this haplogroup is found at high frequency at vast territory from ranging from Iceland to India, okay? So it's found at high frequency in Eastern Europe, also in Northern Europe, also in Central Asia, and also in northern part of Indian Peninsula. Okay, and actually, it's the most frequent haplogroup in Poland. I told you it's almost 60% of males in Poland belong to this haplogroup R1b. Okay. And here is the shortened genetic journey of paternal ancestors of males belonging to haplogroup R1a. The first male from haplogroup R1b was, according to the genetic studies, molecular anthropological studies, was born 10 to 15,000 years ago on steppes of Ukraine or southern Asia, southern Russia, and his descendants are linked to the Indo-European language and domestication of the horse. Okay, so this is the domestication of the horse was the reason why this haplogroup spread on such vast area of Eurasia. Okay, and ancestor of R1a was a male from haplogroup R1, born 30,000 years ago in Central Asia. An ancestor of R1 was a male from haplogroup R. An ancestor of R was a male from haplogroup P, born 35,000 years ago in Central Asia, which is an ancestor of many modern Europeans and Native Americans. Okay, actually, haplogroup P is an ancestor of haplogroup Q, typical for Native Americans, and haplogroup R, typical for many modern Europeans. Okay, an ancestor of P was a male from haplogroup K, who was born 40,000 years ago somewhere in Iran or the southern part of Central Asia. Okay, an ancestor of K was a male from haplogroup F, born 45,000 years ago in Northern Africa or the Middle East, which is an ancestor of the vast majority, 90 to 95% of non-Africans. Okay, an ancestor of F was a male from haplogroup Y, excluding haplogroups A and B, born 31 to 79,000 years ago in Ethiopia or Sudan. Okay, which is who was an ancestor of all non-Africans. Okay, an ancestor of Y, excluding A and B, was genetic Adam living 60,000 years ago in Africa, who was an ancestor of all modern males. Okay, here we can see. So you can see when you detect this haplogroup R1a, you can reconstruct the person's ancestors' genetic journey from where and how were traveling his ancestors to the region where he lives now. Okay, and just to remind you, that just to sum up and tell you something very important, that DNA typing provides only evidence for geographic origin of distant ancestors. These are prehistoric types, and not for ethnic origin, defined by historical types. Because, for example, I am called quite sometimes. I'm called by some people who are interested in such type of anthropological examination. So they would like to know, and because they're asking me questions. They ask me questions. Oh, I would like to prove that I have German origin. Okay. Oh, I would like to prove that I have Jewish origin. Okay. And I'm telling you, no. Okay, I can tell you, I can detect the haplogroup, of course, for example, in your Y chromosome. But this Y chromosome haplogroup does not tell you. It's not a direct evidence for ethnicity. Okay, it tells you where your distant ancestors came from. Okay, but it doesn't tell you anything about ethnicity, because haplogroups occurred in prehistorical times. Yeah, when the modern ethnic groups were not present on the earth. Okay, but of course some haplogroups are specific for some ethnic groups, are specific to only some ethnic groups, and they can serve as additional evidence when the ethnic origin is questioned. Okay, is in question. Okay, and just here is a comparison. Here you can see a phylogenetic tree with different which shows also frequency of an occurrence and the numbers of individuals from different haplogroups in Ashkenazi Jews who used to inhabit Eastern Europe, and in Europeans of non-Jewish ancestry. Okay, and this blue labeled areas is a haplogroups which are found in Ashkenazi Jews, and the white labeled areas these are haplogroups found in non-Jewish Europeans. Okay, and you can see, for example, that if you detect haplogroup J1, which is found in at high frequency as in Semitic populations, Semitic, the Arabic, the Jewish, you can see, okay, the Ashkenazi Jews which inhabited Europe had very high frequency of J1. So, of course, non-Jewish Europeans also have this haplogroup, but it's found at low frequency. Okay, so if you detect J1 and you have, and your question is, is my paternal ancestry was my paternal ancestry Jewish? And if you detect J1 and you have some additional evidence, this will also support your Jewish theory. But if you do not have any additional evidence, the J1 does not indicate that your ancestors were Jewish, of course. And of course, some haplogroups which are typical for Europe, like R1A, which is not found, which is very rare in Semitic populations. Actually, this is also found, you can see, also found in Ashkenazi Jews, which is an evidence for gene flow between Jews and non-Jews in Europe. Okay, let us go further. And here is an example of application. A study, one of the genetic studies concerned Jewish priests, Kohanis, because according to Jewish tradition, the Kohanim could be only paternal descendants of Aaron, who was the brother of Moses. Okay, and actually to this, so this is the Aaron, and here you can see the descendants of this priests are very often carry surnames like Cohen. Leonard Cohen. Maybe you've heard of this singer. And also there is some up there, I think, quite some other American actors or some with this surname. So it's of course of Jewish ancestry. So the question of the researchers was, do people who claim the Kohanim ancestry come from the same paternal ancestor? Okay. So they studied different people, different males, claiming this ancestry, of course, and they found that majority of the Y chromosomes were genetically similar. And they computed the age of the ancestor, the time to the most recent common ancestor of these males. And they found that the common paternal ancestor of these males lived approximately 3,000 years ago, which actually confirmed it, which actually matched the tradition, the information provided, for example, in the Bible, about the Aaron and his descendants. Okay. Another example of application is the haplogroup. Is the haplotype from haplogroup C, which appeared on the Earth approximately 1,000 years ago in Mongolia. Okay, and the researchers found that 8% of males between the Pacific Ocean and the Caspian Sea and which corresponds to 0.5% of males in the world. That is more than 60 million males in the world are descendants of only one male who lived approximately 1,000 years ago. You can see 16 million males are descendants of one man or who lived 1,000 years ago. Do you have any idea who this person was? Who am I talking about? Yes, you are right. Yes, you are right. The researchers, I will write it. Yes, the researchers claim that it must have been Genghis Khan, who, according to historical, who lived more or less 1,000 years ago, and according to historical sources, he had, I think, many children, and his children played an important role in the societies where they live. Okay, and must have such great reproductive success. Okay, let's go further. Another example of application is the genetic study concerning origin of Polynesians. There was Norwegian anthropologist Thor Heyerdahl, who claimed that Polynesians have American origin. These were Native Americans who were sailors and spread in Polynesia, so in the Pacific Ocean. Okay, they settled on the islands of the Pacific Ocean. However, genetic studies show that Heyerdahl was wrong, because the Polynesians actually originate from Melanesia and Asia. Okay, so this is just an example which of genetic analysis which verified and one of the anthropological theories. Okay, and I'm going to tell you about the application of polymorphism of the Y chromosome in phylogenetic studies of the Slavs. Okay. Which is an example of a study which was performed at our department, just to tell you a bit about our small contribution to the topic, okay, to the topic of molecular anthropology. And okay, just to introduce you to the topic, there is a family of languages spoken in the world which is called Indo-European linguistic family. And this linguistic family has the highest number of speakers in the world. Approximately 2.5 billion people in the world speak the Indo-European languages. Okay, and these Indo-European languages are divided into different branches. Okay, they are divided into Slavic languages, Baltic languages, Germanic languages, Celtic languages, Romance languages. And there are some single languages like Albanian, Greek, Armenian, and also another group is Indo-Iranian languages. Okay, so one of these branches, Slavic languages, is a group of closely related languages within the Indo-European linguistic family, and there is approximately 250 million Slavs in the world, which inhabit mainly Eastern and Southeastern Europe and also North of Asia. And Slavs are the most numerous ethno-linguistic group of peoples in Europe, actually. Yeah, and they are divided linguistically and culturally into Eastern Slavs, including Russians, Belarusians, and Ukrainians, into Western Slavs, which includes Poles, Slovaks, Czechs, and also a small ethnic minority of Lusatians or Sorbs inhabiting southeastern Germany. Okay, but they're actually at the edge of extinction. Their languages are becoming extinct very quickly. And the Southern Slavs are divided into Slovenes, Croats, Bosnians, Montenegrins, Serbs, Macedonians, and North Macedonians and Bulgarians. Okay, and what is the origin of the Slavs? So where do Slavs come from? Actually, because you can see, they inhabit a very vast area of Europe. Yeah, huge territory of Europe. So how did it happen that they occupy such a large part of the continent? Okay, so what is the origin? Where they came from? We know that at the turn of the antique times and the Middle Ages, the mass migrations of peoples in Europe took place, and these mass migrations, which were initiated by the invasion of Huns to Europe, resulted in expansion of Slavs to the larger part of Eastern Europe. Okay, and so the first documents which record the presence of Slavs, which we can say they write certain with 100% certainty about Slavs, are sources, written sources from the first half of the 6th century, where a Gothic writer, Jordanes, claimed that had noted that Slavs are a group of it's just a group of people, yeah, which actually we can say that they're almost everywhere. Yeah, actually, the invasion in Europe, the expansion, not invasion, but the expansion in Europe was already highly advanced. Okay, so because of lack of written sources about origin of Slavs, actually it's not known where the Slavs exactly came from, because suddenly they spread to the vast part of the continent, but nobody knows where they came, came exactly. Okay, so there are many hypotheses about territorial origin of Slavs based on archaeological studies, anthropological studies, and also linguistic studies. And the two theories which have gained the highest popularity among researchers are the ones which place the homeland of the Slavs in two regions: in the basin of the Oder and Vistula rivers in present-day Poland, or in the basin of the Middle Dnieper basin, in the basin of the Middle Dnieper River in present-day Ukraine. Okay, in Poland, these two theories are called respectively autochthonous or allochthonous. Okay, so actually this terminology is used only in Poland. Okay, and regarding the first theory, what is the evidence which supports the first theory that the proto-Slavs must have lived in present-day Poland? Okay, now, one of the evidence is the so-called Lusatian archaeological culture, which developed before Christ. Okay, in the basin of the Vistula, Oder, and the Upper Elbe. Okay, and I'm sure that the Polish students know the place of Biskupin in Poland, because they are taught about this place during the history lessons. Okay, this is just an example of the Slovatian culture in Poland. Okay, and so actually nobody knows which ethnic group was responsible for the Slovatian culture. So some researchers claim that it must have been Slavs. Okay, sorry, Slavs. And there are also some Roman sources from the 1st and 2nd centuries who know the presence of Veneti people living along the Vistula River. And for example, some old German name of Slavs is Svenden. Okay, Germans called Slavs Venden. Okay, even I think in the until now in Germany, the lower Sorbian speakers in Germany until now are called that they are Wendish people. Okay. And there's also, I think, until 19th century, Slovenes in the Alps were called by the German speakers Winden. Okay, so you can see. And generally in the also, I think, according to some Icelandic documents, the first ruler of Poland, Mieszko I, was called King of Wends. Okay, actually, yeah. So this is why. So some people claim, oh, because Slavs were called by Germans, by the Germanic people were called Vents. Yeah, so Veneti must have been Slavs. Okay. Also, Jordanes, the writer which I told you about, also claims that Veneti is something which is equal to Slavs. Okay. So what is the evidence which supports the other theory? The theory that the homeland of Slavs was located at the Dnieper River. So the first evidence comes from comparative linguistics, which tells us about time and place of social and cultural contacts with other languages. Okay, because of this, because we can say that we can reconstruct, linguists can reconstruct that different words in Slavic languages came from Baltic languages, from Germanic languages, or Iranian languages. Okay, and for example, there are quite many words in the Slavic languages which come from Iranian languages. For example, there is a Slavic word Bóg in Polish, yeah, which means God, yeah, and actually this is a word of Iranian origin. Okay, so it is assumed that Slavs must have lived close to the Iranian language speakers. Okay, and we know from historical sources that some Iranian speakers lived at the seashore of the Black Sea. Okay, so it's more likely that Slavs must have lived at the Dnieper than at the Vistula and Oder, because Vistula and Oder are rivers far away from the Black Sea, okay, and from the Iranian speakers, okay. And another evidence is that the Slavs do not have a native name for the beech tree, and also for several other plants. So actually the beech tree is in Polish language is called Bóg. Okay, this is the tree. Okay, and according to the linguists, this word was borrowed from Germanic languages. Okay, so because Slavs do not have a native name for this tree, they must have lived in an area where these beech trees were not present. Okay, and in Poland there are many beech trees, and in Ukraine there are no such trees. Yeah, so this is another evidence. Okay, and also another hypothesis is that some tribes of Neuri and Budini living in the Dnieper basin, mentioned by Herodotus in the 5th century before Christ, were actually Slavs. Okay, but this is of course a theory, and it cannot be proved. Okay, and I told you that population studies show significant differences in distribution of Y chromosomes even between closely related populations, which suggests that the Y chromosome may be particularly useful in studies of genetic studies of relatively recent historical events. Moreover, the Y chromosomal studies show that the Polish population is homogeneous, and there are significant differences between Polish and neighboring non-Slavic populations. Non-Slavic populations are German and Lithuanian populations. Okay, and actually, because the Slavic expansion in Europe is a relatively recent event, was a relatively recent event in the history of our continent. Okay, and the Y chromosomes differ so much between different human populations, even closely related populations. We assume that our department that they can be suitable for the study of the origin of the Slavs. Okay, and here is a study of different European populations from 2001, quite old, yeah, you can see, and there were only 3 populations of Slavic populations studied then, yeah, populations of Warsaw and Wrocław in Poland and population of Moscow, and they found that these 3 Slavic populations formed a separate branch in the phylogenetic tree of European populations, okay? And we, at our department, we started comparing Y chromosomes of different Slavs, and we found that many Slavic populations, including Eastern Slavic Belarusians and Russians, including Western Slavic Slovaks, and also Southern Slavic Slovenes, were genetically indistinguishable from Ukrainians. Okay, that many Slavic groups were genetically identical, homogeneous as Ukrainians, whereas the Polish population was genetically similar to the Ukrainians, but was actually a separate population, was genetically distinct from the rest. Okay, and you can see here just the number of populations which showed homogeneity, genetic similarity, yeah, in comparison to Polish and Ukrainian populations, just assuming that homogeneity means that the p-value was higher than 0.05. Okay, so we can see, oh, for the Polish population, only one, but for the Ukrainian populations, there were populations of Western Slavic origin. There were Western Slavic populations of Poland and Slovakia, Southern Slavic population of Slovenia, and many Eastern Slavic populations. Okay, based on these results, we actually we concluded that it is much more likely that the homeland of Slavs was in present-day Ukraine than in present-day Poland. Okay, because if origin of present-day if the homeland of modern Slavs had been in present-day Poland, there must have been more. There should be more Slavic groups genetically similar to the Poles. Okay, but actually our genetic studies did not find such a phenomenon. So actually, we published this results in the Journal of Human Genetics, which as the first known example of application of genetic markers in an attempt to answer the question about the origin of Slavs. And I can tell you, it's still very well cited paper of our paper. So just to tell you an example of the application of the genetic analysis of the history of modern human populations. Okay. Now I'm going to tell you a bit about the genographic project, which was a project which I took part personally. A huge international, initially it was a huge international project of molecular anthropological studies covering the whole world, which started in 2005. Initial period was 5 years, and and this time was extended further, and hundreds of thousands of people from native ethnic groups of the whole world were studied, and there were 10 leading research centers in the world, so that the world was divided into different parts. So, for example, different centers were responsible for different parts of the world. Okay, and the aim of the genographic project was to discover genetic differentiation of native human population, the whole world, to reconstruct the human genetic journey and discover time and paths of prehistoric and historical migrations, as well as time and paths of colonization of individual continents. Okay, and I will go through it very quickly, but I will tell you that I was a person who took part in this project and also published an interesting journal paper entitled Contemporary Paternal Genetic Landscape of Polish and German Population from Early Medieval Slavic Expansion to Post-World War II Resettlements. Just another contribution of mine to the origin of the modern Slavs. Okay, so thank you very much. Let's have right now 5-minute breaks. Okay, only 5 minutes, and then we will continue with another topic. Okay, so let's meet at 20 past 9. Okay. Okay, this is an example from our practice. There was a paternity testing case in which we analyzed serological markers. And in one of the markers, which is acid phosphatase, which is an enzyme found inside red blood cells, the following situation was found, that the putative father was BA. Okay, please do not look at this D letter. Okay, the putative father was BA, the mother was A, and the child was B. Okay, so we know. Okay, so this was actually analyzed by one of departments of forensic medicine in Poland, not in Gdańsk, and they found that the child inherited B from the father because the child was B, and but the mother was A, the child was B, and nothing was inherited from the mother. So the question of the court was, why is the tested man the father of the child? And the answer from the laboratory came, the mother is not the mother of the child. Okay, so the court was shocked, the judge was shocked, what's going on, why, come on. So they sent this case to our department for examination, and we found what we found in our laboratory was that this woman and this child were not homozygous AA and BB, but they were heterozygous, having a very short allele which is called D. Okay, so this is just an example of this null allele, the silent allele. Okay, actually we thought that these two people were the other laboratory in Poland thought that these two people were not related because they had nothing in common. But please note that they were two opposite homozygotes. Yeah, they were AA and BB. And our study showed, our examination showed that they were not homozygotes. They were false homozygotes. They contained allele. They carried allele D, which is not routinely detected in standard serological examination. Okay, just to make you, because this allele D was quite interesting to us. We included into the examination all the sisters of this woman and her mother. However, none of these women carried this allele D. Probably it must have been inherited from a deceased father. Okay, another case with the acid phosphatase, just to show you that there are more such cases. The mother and that there was a family in which we found an allele which is called R. This is also another rare allele. Okay, and here is an example of our report on examination performed, the genetic examination performed in paternity tests. Okay, what is interesting and important in this report is that the people who were tested came to us simultaneously on the same day, the same hour. Okay, the defendant, the mother, and the child arrived together. Okay, my question to you is, why do you think it is recommended that all the people taking part in paternity testing come together? The same day, the same hour. Come on, I would not trust an expert, a forensic expert, who claims, oh, please come together because there is a risk that I will make a mistake, an error if you come separately. Come on, no, it is something. I would call this person totally unreliable. No, it's not an expert. It's a person who makes mistakes. No, come on, some other reason. What is the reason? What do you think? We want them to come together on the same day, the same hour. And it's not a matter of an error. Mm-hmm. Come on, this is just a guessing game. You do not have to be correct. Just
maybe to see them and compare how they look like, and to make some propositions about can they be relatives
And for example, we can make some anthropological comparisons, yeah, whether they are similar to each other. Yeah, do you mean this?
Like hair, eyes.
Yeah, we could do it. Yeah, but this is not the thing that court asks us, yeah, about for. Of course, it is possible to use anthropological examination for paternity testing, but people who come to us, they come only for genetic analysis of genetic markers. Yeah, of course, we just make such a guessing game. Oh, it's for me, I'm sure that he's a father because he's so similar. Okay, and then or some other hypothesis. Oh, I think that he's not the father. Come on, this child has, I don't know, black eyes, and none of the parents have black eyes. So they are just like, this is just our guessing game, yeah, of the people. But no, this is not the reason why we want them to come together. Because we could also just make photographical documentation. Yeah, they do not. But something which is important, why we would like them to come to. Okay, now let's imagine that the mother arrives on Monday. Okay, so let's imagine that the trio, the mother, the child, and the defendant, were invited to come to us on Monday. However, only the mother and the child come. Okay, so we collect samples from them, and the man could not come on Monday. And he arrives on Tuesday, and we collect the samples from him on Tuesday. Yes, you are right. Yeah, exactly. So we call it confrontation of the parties. Yeah, the woman confirms. Yeah, she can see who from whom we collect the sample. Yeah, and she confirms. Yes, this is this man in the case. Yeah. And this similar situation, the man can see, oh, this is this woman, yeah, and this is this child, and no other child, yeah. So this is, yeah, we call it confrontation of the parties. However, if the two parties agree to have the samples collected separately, we also agree, yeah. But then we take the photos of these people. Yeah, we take the photos. So if they come together, we do not take any photos. But if they come separately, for example, on Monday and Tuesday, yeah, then we take the photos of the people, and then we attach this photos to documentation, and send to the court. Yeah, just as evidence who came to us for sample collection. Okay. And then we also ask, before the collection of the sample, we ask also several questions to these people. Okay, the first question is about blood transfusion during the last 3 months, because if there was blood transfusion, there can be some foreign antigens, yeah, some foreign proteins of another person, and you know that 3 months are sufficient to exchange red blood cells, yeah, totally. Yeah. So this is something which we asked about, which is quite important. Okay, and another question. So I told you, we asked 3 questions. So the first question is about blood transfusion during last 3 months. Second question: what would you ask? What would you ask about in the second question? Exactly. Yeah, this is what we ask about. Has any one of you had a bone marrow transplant? Yeah, this is something which is important, because if a person, for example, if a person, if a putative father is a bone marrow recipient, can we perform blood group typing for paternity testing? What do you think? Can we perform blood group typing for paternity testing if we have if a putative father is a bone marrow recipient, underwent bone marrow transplant? Victor, yes, you are the person who is speaking. Why don't you think so? Yes, yes, yes. That can be. Yes, I agree with you fully. Yes, so this is something. Actually, this person is totally actually is excluded for blood group typing for paternity testing, yeah, because there be some foreign antigens present and so on. Okay, so this is the first thing. So if we have, so what should we collect from this person? So sorry, another question. Can we perform DNA typing for paternity testing when we have a bone marrow recipient as a putative father? Yes. What would you collect?
Hair?
Lizaveta, yes?
Yes.
Okay. Yes, her. Lisa. Okay. Yes, you are right, actually. What we collect for, because some of you would think of buccal swabs, yeah, because it is very convenient to collect buccal swabs, yeah. It's non-invasive and very simple. However, studies show that bone marrow recipients have mixed DNA profile in the buccal swabs. In the buccal swabs, there is mixed DNA. There are mixed DNA profiles of a donor and the recipient. Okay, so this is why it is quite difficult to analyze such profiles, because like there are two individuals mixed in this buccal swab. Okay, so this is why it is recommended to collect hair bulbs, yeah, hair follicles, because in hair follicles there is only there's extremely small, but there is extremely small amount of DNA from the donor. Okay, so this is, yeah, so I agree with you, Lizaveta. This is something which if we know that this person is a bone marrow recipient, we collect a hair follicle. Okay, a hair follicles. And the third question which we ask about, I will tell you about this question later. Okay, and here is an example of no exclusion. Okay, this is the putative, just as you did during your exercises, yeah, last time, yeah, 1 week ago. Okay, so this is a defendant, this is the mother, the child. In all the serological markers shown here, the child has something in common with the mother and with the defendant. So the conclusion is that the paternity cannot be excluded. Okay, you remember, we do not call it that, we do not write that he is the father, that he's likely the father. No, we write that paternity cannot be excluded. And here is an example of exclusion. The child is B at the ABO system. The B did not come from the mother, and the B did not come from the defendant, so it must have come from the real father of the child. Okay, and I don't remember, there are more. There is also, there is another exclusion. The acid phosphatase, the child is B. It means homozygous BB. One of these B's came from the mother. Second one could not come from this man because the man is CA. Okay, so please know that there are no opposite homozygotes with a possible silent islet. No, this is just the only homozygous individual is the child. Okay, and another exclusion, PGM1. Please look at the results before the brackets. Results of electrophoresis. The child is 2-2. One of these alleles 2 came from the mother, and second one could not come from this man because the man is 1-1. Okay, and also results in the brackets for isoelectrofocusing. The child is 2+2+. One of these alleles came from the mother, and second one could not come from this man because the man is 1+2+. So there we have 3 markers which show that in which we found alleles which are not paternal and are not maternal. So the final conclusion is that the man is not the father, of course. Okay. So what can be the source of DNA regarding forensic genetic examination in paternity testing? Of course, it can be each cell of the organism which contain a nucleus. So my question to you, so actually these are all cells of the organism except for several cells. So what cells do not contain nuclei? Do you remember? Yes, erythrocytes and platelets. Actually, platelets are not cells. They are just like fragments of cells. You are right, they do not contain nuclei, but I would not call it cells. They are just like fragments of cells, yeah? And I told you about some type of biological material which does not contain nuclei and it's only suitable for mitochondrial DNA analysis. So, what material am I talking about? Which is suitable only for mitochondrial DNA analysis in forensic genetics, because this material does not have nuclei, and only mitochondrial DNA is tested when we have something like this. Just, you remember, maybe you had the course of histology during your studies. Just remember, there are some cells which also lose nuclei and when they become mature, and they even become dead when they become mature. Which cells become
Epithelial
dead?
cells?
Pardon?
Epithelial?
Epithelial cells, yeah. What's the name of the cells? Elizaveta, yes? Elizaveta, okay.
Yes.
What's the name of the cells in histology? Remember, there's a protein which is abundant in the cells. What is the protein?
Keratinocytes.
Yes, exactly. Okay, so these are keratinocytes. So what the material which I'm talking about is a hair without root, without a root. Okay, so if we have a hair which is cut with scissors without the root, actually there is no nuclear DNA. There are only keratinocytes. So actually, the way how such a hair is processed is that this nuclear DNA is not analyzed, only the mitochondrial DNA. Okay, let us go further. So, and of course, this can be blood, which contain. Of course, you told me about erythrocytes, but of course, it's difficult to. It's not possible to collect the blood sample or to leave a blood stain without nucleated white blood cells. Okay, so of course, blood is a very good source of DNA. Unless we have, for example, pure red blood cell mass for transfusion purposes, there will be no, there shouldn't be any nucleated cells. Okay, this can be also semen, saliva, urine, hairs without roots. I told you, but hairs with follicles are okay. Teeth, bones, soft tissues, archival material. This is what we use for genetic analysis, forensic medicine. Okay, and this slide is just to tell you that everyone has a unique DNA profile. There's no one else in the world who has identical DNA profile as mine. Am I right? Am I right? Or is there someone who is identical as me, who has identical DNA profile as mine?
Only if you haven't a clone.
Pardon? If I have a whom?
If you have clone, then yes.
Clone. No, I don't have a clone. But this is a good idea. I don't have a clone. But what clone are you talking about? There is someone on the chat. Let me read. Clones. Yes, but generally humans are not cloned. I'm not sure if there was twins. Yeah, Belen, you are right. Yeah. I will write your answer. Especially the monos, actually not all twins, not because dizygotic twins are different, but we are talking about the monozygotic twins. Yeah, so of course they are genetically identical as me. But I do not have a twin brother, so I am unique. There is no one else in the world who has identical DNA profile as mine. Okay, so this is something. So the third question that we ask about in the form during the collection of a sample from a person for paternity testing is a question asked only to the putative father. Do you have a twin brother? And if he says yes, then we ask, do you know whether he is a monozygotic or a dizygotic twin brother? Okay, because if he has a monozygotic twin brother, then the techniques of STR typing used routinely in forensic genetics are not able to distinguish who of the two men are brothers. Okay, so we are not so in order to, so this is why we ask. So if there is a suspicion that we do not know who of the two twins is the father of the child, then maybe some just we always because we know that, for example, the putative father has a monozygotic twin brother, then we write in the final conclusion that just because of the fact that the monozygotic twins are genetically identical, this answer doesn't, our results do not answer who of them is the father of the child. Okay, so just to remind you that that this is the just to tell you that this is the third question that we ask during the collection of the sample. We ask also only to the putative father. Okay, let us go further. Here you can see genetic fingerprints obtained with the use of multi-locus probes for 4 pairs of twins, and you can see pairs B and C are monozygotic twins. Okay, pairs A and D, these are dizygotic twins. They are similar, but they are different. Yeah, but pairs B and they are genetically identical. Okay, and now we have a science fiction question, because some of you suggested to me, oh, clones are genetically identical. How to tell it? Because okay, so if there is a, is it possible to distinguish? So if there is, how to ask you this question? Okay, now let's imagine that we do not know who of 2 monozygotic twin brothers is the father of a child. Is it possible to do something to distinguish who of them is the father of the child? Is it possible, do you think? Methylation, but in methylation is not inherited. Yeah, something which one of them is a father of a child, and who of them is the father? Forensic STR typing shows that both are fathers of the child. Yeah, with 99.9999999% both are fathers of the child. But what to do? Piętnowanie. Yes, there is something which is responsible for some genetic disorders, genetic imprinting, it's called. Yeah. Some, it's also, it can be inherited, but it, and it also, it's maybe, how to tell you, yeah, but regarding whether it is detected, for example, later in the epithelial cells or in blood, no, I think it wouldn't be during the development of embryo. Yeah, it may matter. Yeah, and there are some, like how to say, uniparental dysomy disorder, genetic disorder, that both chromosomes come from only one parent. So there is a different methylation pattern, and it matters during the development of the fetus. But I think when we have a child who is born, and come on, and I also guess that it's only I think it's only the matter of the gender and not the, because I think, okay, both male brothers have the same gender, the same sex. Yeah, they are males. So the imprinting will be, I guess, identical. So no, this is not the. I like that you think and that you try to find the answer, but let's try to find the correct answer. So thank you very much, Victor, for your answers. I like them a lot.
Maybe we can differentiate them by some specific point mutations.
Yes, very well. Yes, that will be for sure, Camila. For sure, there are some somatic mutations which occurred only in one of the twins during the development of the fetus. Yeah, of the fetus, and maybe during some early childhood, during development of sperm cells or the testes. Yeah, the testes and the sperm cells or something like this. So how to or during the fetal development. So how can we detect these mutations? What should we do? Which technique should we use to detect them? What would you recommend to do?
Probably sequencing.
Pardon?
Probably sequencing.
What kind of sequencing?
I didn't say what kind of sequencing. I said only sequencing.
Sequencing, yeah. But to sequence what? What would you sequence?
Whole genome.
Yes. Who told it?
Elizabeth.
Elisabetta. Okay. Whole genome, yes. But how would you sequence whole genome? Yeah. Actually, yeah, you are whole genome sequencing will detect differences between the two twins. Probably quite many sequences, many somatic mutations, and actually, I know one published paper on paternity testing of a child, and two twin paternal and two putative fathers who were twin brothers, yeah, and monozygotic twin brothers, and this whole genome sequencing found somatic mutations in one of the twins who were inherited by the child. Okay, so it was possible to distinguish who of them is the father of the child. But of course, whole genome sequencing is not used routinely in forensic genetics, and actually it's not so it's something which is still very costly and is not used routinely. But just to tell you that it is possible to answer also this question, okay, about paternity of monozygotic twin brothers. Okay, another question. This will be going to be a science fiction question, because you told me about this. You told me the answers of that clones will be identical as me. Yeah, I agree with you. Clones will be identical as me. Okay, so now let's imagine that somebody cloned me, and my clone is a criminal and commits a lot of crimes, okay? And the DNA sample was found in the crime scene, and the DNA profile indicates that it was me who committed the crime, okay? And I'm in the court and I'm telling, no, it was not me. It was my clone. Somebody cloned me. It was not me, really. I know that somebody cloned me, and my clone is a bad person who commits crimes. Yeah, just like this is a science fiction story. But now that question, and now I have a question to you as biotechnologists. Yeah, how would you? There's something on the chat. We can take a look. Yes, David, you are right. Maybe we can take a look at epigenetic changes, David. This is a good. How can we just? So the question was already answered. My question, you do not know the question, but David already answered that. Maybe that the question, of course, was that how can we distinguish who committed the crime? That it was. How can we prove that it was not me but my clone? And one of the ways how we can do it is to detect epigenetic changes. Yeah, of course, this is very good. So. What else could we do? Yes, Victor, it was you? Yes? Okay, yes, you are right, because cloning, yeah, only removes the nucleus and the mitochondrial less, so we can compare the mitochondrial DNA, and it's quite likely that the egg cell to which the nucleus, a new nucleus was introduced, actually came from another maternal lineage, so it is possible to detect it, okay? Okay, so very well. And another question. Now let's imagine. So okay. So you told me the answers if we have a DNA from a sample. Okay. And now let's imagine that we have a blood sample. What can we analyze in this blood sample in order to answer the question? Who of the two twins, like monozygotic twins, the sample came from? Okay, we have like a crime committed by one of two twins, and each of the two twins claims it is not my. This is my sample. This is my blood sample, which is an evidence that like alibi, yeah, because I did not commit this crime because I was in a laboratory. A blood sample was collected from me. And this is evidence. This is blood sample collected from me during when I was in laboratory. So I could not murder this person. And the other brother says, no, it is my blood sample. I was in the laboratory. So how can we, what can we check in this blood in order to answer the question from whom this blood was collected? Of course, we can test DNA. You are right that we can test DNA and check the epigenetic changes. We can sequence their whole genomes or something like this. But something more simple. What can we check in this blood sample? What can we analyze in this blood sample to distinguish from whom of two monozygotic twins this blood was collected?
Level of sugar or something like that, because if they had different diets, then they will have a different amount of such things.
Yeah, it is possible that they have, you mean like the, how do we call it correctly in English, glycolated hemoglobin, yeah? The one which is related to diabetes, yeah? Yeah, but this is actually, so if they are healthy and they do not suffer from diabetes, I guess we will not detect it, yeah? We will not detect this glycolated hemoglobin because, but I would like to to find the question, to find an answer which will be universal to all situations. That of course we can say, oh, one has HIV and the other does not have, yeah? Maybe one takes some drugs, or maybe one, I don't know, works in a factory and we can find some compounds from this factory in his blood or something like this. But let's think of an answer which will be universal for all situations. No diabetes, no viral infection, like chronic infection or What can be different in bloods of two genetically identical individuals? It must be something which is a result of interaction with the environment. Okay, let's look at chat. Searching different, yes, methylation status will be different, you are right, but I'm asking about something which is not related to DNA analysis. Yeah, but these are parameters we actually, they can be totally different into twins, yeah, you are right, but they change. Yeah, because we do not, let's say that the sample was collected, let's say 1 month ago, and maybe a lot of things could have changed in the blood, yeah, another factors which depend on diet like lipidograms, yeah. Okay, but the antibodies, Camila, you are right, yeah, antibody profiles will be different, yeah, they can be, because there are different, there can be different infections, different vaccinations. Even the same antigen can trigger totally different immune response. Yeah, and so this is something which can be, which is suitable for all situations. Like lipidogram, yeah, it depends on what they ate on the date when the sample was collected or something like this. Yeah, but antibody profile, this is a very good answer. Okay, and of course, if we have what is another thing which makes monozygotic twins different? Do you know? Let's assume that we have two cadavers of monozygotic twins. Yeah, and they lie in our autopsy room. What is the first thing that can be checked? Who of them is who? Without any genetic analysis of DNA or something like this. What do teeth? Yeah, you are right, teeth. Victor, you are right. Yeah, but something. Yeah, so the odontological examination can be performed. You are right, but something else. Something. What can be checked? What can be analyzed in a criminalistic laboratory? not DNA. What do monozygotic twins differ in? Do you have any idea? It's something which, when you think of criminalistic examination, many of you think of DNA, but some other people would think of something else. What else? Okay, let's look at chat. Tattoo scars? Yeah, you are right. Yes, that is okay. But another Belen, yes, Belen, you are right. I agree with you. Fingerprints, yeah, then they have different fingerprints. Yeah, so this is something which you should remember. And monozygotic twins have the same DNA in general, but they have different fingerprints. So this is so Belen is right. Okay, thank you very much. And tattoo scars is also a good answer. Let's go. But this is a special situation. They have that one of them has these tattoos and the one does not have. Let's go further. And we can see here a genetic fingerprints of a mother, a child, and two men, F1 and F2. One of these two men is the father of the child. Which is the father of child C? Who is the father? F1 or F2? F2 is the father. Yes. Why? F2, yes, you are right. F2, why is F2 the father? Some explanation.
Because we can see the bands that are not from the mother in the identical place that comes from the F2.
Exactly, yes, you are right. Yes, exactly. The bands in the child which are not maternal, I like what you said, which are not maternal, which did not come from the mother, are found in the father. Yeah, we look at the child. Okay, this band is found in the mother, so it's likely maternal, but this one does not come from the mother and is found in F2. Okay, this is how we compare these genetic fingerprints. And here you can see genetic fingerprints of a blood stain found in the crime scene, yeah, and different suspects, and you can see that suspect number 3 has a DNA profile which is a genetic fingerprint which is identical as the genetic fingerprint of the blood stain. Okay, so these are just genetic fingerprints obtained with the use of RFLP analysis and multi-locus probes. Okay, let us go further. And okay, and here you can see result of for a single locus probe. Okay, every person has always maximally 2 bands. One comes always from a mother and the other one comes always from a father. Okay, so here you can see an example of paternity exclusion. Here is a band in the child which is maternal. This one does not come from the mother and is not found in the alleged father. Okay, and here you can see the same. The opposite situation, yeah, this man is child found in the mother, and this one matches the alleged father. Okay, let us go further. And here is another example from 2002, just to show you how this examination looks like. And the bands are, I think there is no exclusion here. But what is important is that we always provide the name of the enzyme which was used for digestion and the aim of the molecular probe, because if any other laboratory uses the same enzyme and the same molecular probe, they should see the same pattern, the same sizes of the fragments. Okay, then the sizes of the fragments are computed by the software by comparison of to the size standard. Okay, so they are not exact sizes, but they are simply computed by the software. Okay, and here's a question to you. This is a task for you. We have results of single locus probe genotypic of a man, a woman, and their 4 children. And one of these 4 children is not the biological child of this couple, this mother and this father. And my question to you is, which child is not the biological child of this couple? Okay, let me see. Second child.
Child 2.
Child 2. Okay,
Because
but and explanation why child number 2?
Because no bands are both from mother and father. Only they are identical with the mother, but not with the father.
Yeah, this is Małgorzata who is speaking. Yes. Okay. Yes, I agree with you. Can you see child number 2 has nothing in common with the father? Yeah. This band is shared with the mother. This band is shared with the mother, and nothing in common with the father. So this is the correct question. This is the child who is not the child, biological child of this couple, this mother and this father. Another question, which is actually more difficult. We have results of single locus progenotyping of a man and his 4 children. Okay, and the question to you is, where is the father? Okay, so we have 5 people. 1, 2, 3, 4, 5. These are 5 people, and one of these 5 people is a father of the rest. So my question to you is, where is the father? This is like an IQ test, actually. It's a task from an IQ test.
Number
The
3,
third one.
Number 3. Why number 3?
because all of the bands are covering with the rest.
Pardon, once more, could you repeat?
All of bands are covering with the rest of
Yes,
people.
yes. Camila, yes, am I right?
Yes.
Okay. Yes, there are some other answers on the chat. Yeah, I can see you are right, but it's quite for you. It's more difficult to explain why. Okay, because you guessed it. Yeah, but for me, it's important that the person explained why. Okay, so yes, you are right. So the only person who can be the father of the rest is person number 3, because every person has either this band or this band. Yeah. For example, person number 1 could be the father of all the people except for person number 4, because they have nothing in common. Okay, great. So let's continue. And here are results for STR markers. Yeah, for an STR marker, this is an analytic ladder. The markers, the alleles are separated by jelly electrophoresis. So this is the same situation. Yeah, this band in the child is paternal and is not found in the putative father. And an example of lack of exclusion, this band is paternal in the child and is found also in the putative father. Okay, and here is an example of results of multiplex PCR and amplification of STR markers with primers labeled with different fluorescent dyes. Okay, so you can see there are 2 individuals here. They have different gender. Yeah, one of them is a male, and the other one is a female. Do you know the name of a gender marker which is used commonly in forensic genetics? Probably I did not tell you, but maybe some of you know it. Okay, the name of the marker, the gender marker, which is used most commonly in the whole world in forensic genetics, is amelogenic. Okay, this is amelogenic. One of these 2 individuals has 2 peaks at amelogenin, and another person has 1 peak at amelogenin. Okay, so now look at amelogenin and tell me where is a male and where is a female, and why? Okay, because I just this is something which I appreciate the most. That you are claiming that that this is a female because there are 2 peaks and they are located on the X chromosome. Okay, this was Victor. Yeah, I can tell you, no, you are. This is a male. This is a male and this is a female. Okay, and now why is the upper person a male and the lower person is a female? Do you have any idea? This is a male. 2 peaks, a male. 1 peak, a female. You're like a smart group, and I'm almost sure that you will guess it.
When 2 peaks, it means that it on both X chromosomes.
Could you please repeat? Maybe
It's
you're
like,
right.
2 X chromosomes, and then 2 peaks.
2 peaks means what? 2 chromosomes?
X chromosomes.
Like 2 peaks means 2 X chromosomes. But this is a male. And this is a female.
Okay.
This
So
is a
maybe
male.
because that 2x will have the same, like the peak will be like nałożyć się na siebie. I don't know how to say it in English.
That Małgorzata is speaking. Yeah.
Yes.
Say it from the beginning. Yeah. Please explain, because you are right. I'm sure that you are right. Yes.
The lower one is a female because we can only see one peak because the brakuje mi tego słowa po angielsku. Że te piki...
Cover, we can say cover.
Okay, so the peaks from both X will cover each other, and
yes.
on the first one we see different peaks because one goes from X and one goes from Y, and that's why
yes,
they are different
right.
chromosomes.
Great, you are yes, you are fully right. Okay, Małgorzata, yes, I'm just. You are right. So the Y, there was an answer on the chat. Because the gene is an autosomic chromosome. No, it's not located on the autosome. It's not an autosomal marker. Yes, the amylogenin gene is located on the X and Y chromosomes. Yeah, but they have different. The sequences on the Y and X chromosomes differ. So the version on the the X chromosome, it has a deletion of 6 nucleotides. Okay, so when you see and the version on the Y chromosome has insertion of 6 nucleotides. Okay, so the longer product is from the Y chromosome and the shorter product is from the X chromosome. And whereas the female person is like homozygous individual. Yeah. Like only one product is amplified from both identical X chromosome. Okay, so this is how forensic geneticists routinely decipher the sex of an individual. Okay, so we can see we genotyped in total 10 STR markers in one reaction, plus amylogeneous agenda marker. In one reaction very quickly. Okay, so we required several hours for extraction of DNA, several hours for PCR, maybe 1 hour for electrophoresis. So, and we obtained DNA profiles which are found once per 3 trillion people. This is trillion in English. Okay, once per 3 billion in Polish. It was found once in Spanish, a billion in Polish and Spanish. Okay, it's once per 3 trillion people. Okay, so you can see that if we are in a hurry and we can prove in less than 1 day that this is a person who committed whose DNA was found in the crime scene. Okay, so we can see that they are. This is such a rapid and quick technique. According to the regulations, according to the Polish law, a person can be arrested without the verdict of a court for maximally 48 hours. And thanks to this multiplex PCR, we can prove very quickly that this was a person somehow related to the crime. Okay, and an interesting story which I would like to tell you about is known in the literature as a case of Phantom of Heilbronn. This case concerned a serial female murderer who was murdering regularly for 16 years in 3 European countries, in Austria, France, and Germany. Okay, so genetic examination was providing evidence that it was always the same woman committing these crimes. Since she was not identified for many years, she was called a woman without a face. There was even some suspicion, I think based on the mitochondrial DNA examination, that it was a woman of Eastern European origin, because some haplogroups, probably haplogroup indicated. Okay, but nobody knew who she was, and it was. I can tell you, and finally they managed to solve this case. They found that in all these cases committed by this woman, the policemen, technicians, they were collecting biological stains with the use of swabs. Yeah, okay. I have a swab, and I'm using it to collect the swap this camera, yeah, and this swabs used in which the swabs used in these crimes in which DNA of this woman was detected were produced in one factory. In this factory, there were working women from Eastern Europe. One of these women was regularly contaminating freshly produced swabs with her DNA. I don't know what she was doing. Maybe she was cleaning her ears and putting back to boxes, yeah? But you can see that for 16 years, police of 3 European countries was looking for a person who was totally innocent. Moreover, there was no serial murder at all. All these murders were committed by totally different people. And the only thing in common was the profile from the factory. Yeah, you can see how dangerous PCR technique can be, because there were probably minute amounts of DNA transferred from the factory, but they were sufficient because PCR is so sensitive. They were sufficient to detect DNA profile, complete DNA profile of this woman, who was already in the database as a murderer. Yeah, so this is something which, so just to make you aware of. Okay. Now I would like to tell you, because we wouldn't like to have such a situation that every laboratory uses its own set of markers in forensic for forensic purposes. Okay, this is why there are markers which are like the markers of first choice, yeah, which are recommended to be analyzed at the beginning, so that all the results can be compared between different laboratories, yeah, that that maybe it can be used for laboratory comparisons, yeah, that just to provide some documents, some certificates that the laboratory works well, yeah. We take part regularly in such comparisons organized by the International Society for Forensic Genetics. We get some samples, I think, from Norway, and they send us blood samples, and they want us to determine DNA profile, and we always pass this comparison, this test, and we always get a certificate that our results are reliable. So we take part in this. So it's just suitable for also to, sometimes people do not agree with the results, so they can make another examination in another laboratory, and the results should always be the same. So it's always very suitable just to compare the results and be obtained between different laboratories. Okay, so where do names of these markers come from? Some names of the markers come from the genes within they are located. For example, VWA is a marker which is located, STR marker, which is located within the intron, within the intron of a gene encoding von Willebrand factor. Okay, but right now, majority of STR markers used in forensic genetics are located far away from any genes. For example, this marker. Okay, and my question to you is, why is this marker called so? Do you have any idea? Such D21S11. What do you think is the origin for the name of this marker? You should guess. Exactly, you are right. You have so many. You are so active, Victor. Okay, that's good because it matters. Yes, you are right. After the D letter, we can see a chromosomal number. Yeah, so D21 means a marker located on chromosome 21. D18 means a marker located on chromosome 18. DY means a marker located on chromosome Y. Okay, DXY is a marker located on both X and Y chromosomes. Okay, and S11. After the S letter, we can see a sequential number which is given to the markers to make so that they have different names. Okay, so it's only a sequential number so that markers have different names. Okay, so this is how they. Okay, and here is a story concerning genetic identification of remains of the royal family of Russia, because I don't remember, but if I don't know if you remember from your schools that in 1917 there were two revolutions in Russia. During one of them, the royal family of Russia was murdered. This included and according to historical sources, 11 people were murdered: the Tsar, his wife, 5 children, and 4 people who was always accompanying the family, like servants. Yeah, I know we can call them servants, but they were not like fully servants, because they were, for example, also a medical doctor who was always accompanying the family, because the Tsar's only son was suffering from hemophilia, whose gene was transferred from the British royal family, because Tsarina Alexandra was from the British royal family. Okay, so and actually nobody knew what happened with the bodies. Yeah, in 1980s. In the Ural Mountains in central Russia, just on the border of Europe and Asia, nearby the city of Yekaterinburg. Then it was called Sverdlovsk. Yeah, right now it is called Yekaterinburg. They found human remains were found, and they found that there were, and they discovered that there were only remains of 9 individuals. Okay, not 11, but only 9. So they started comparing DNA profiles of these individuals, and they found that 3 individuals could be daughters of one couple, and the remaining 4 people were unrelated. So these 4 people could have been servants. Yeah, let's call them servants. Yeah, and one of the daughters and the only son were missing. Yeah, and there were even some suspicions that maybe they survived the execution. But right now we know they did not. There was even, I know a story of a woman who was claiming to be the Tsar's only surviving daughter, Princess Anastasia. Yeah. But now we know that she was lying here, because the daughter of the remains of the last daughter and the son were found later, and so they did not survive. Okay. And another story from our area concerning genetic identification of the remains of the famous Polish astronomer Nicolaus Copernicus. I don't know how many years ago, but not so long ago, not so long time ago, some human remains were found in the Cathedral of Frombork. Frombork is a town quite nearby from here, to the east from here. And there was a lot of evidence that Copernicus was buried in this grave. Okay, so because there was a lot of evidence, but also forensic geneticists were asked to perform examination. So what did they do? In Sweden, they found a book which belonged to Nicolaus Copernicus. Okay, this book was robbed from Poland during the Polish-Swedish War, I think of the 17th century. I don't remember exactly. This war was called Potop in Polish. It means flood. Yeah, flood of Swedes to Poland. Yeah. Where during this war, a lot of stuff was taken from Poland, and the Swedish people did not give it back until now. And in one of these. So in this book, which belonged to Copernicus, they found a total of 9 hairs, and geneticists determined the DNA profile of 3 hairs from the book with DNA profile of the remains from the cathedral, and they found that they matched perfect. So actually, they proved it was Copernicus, but Copernicus was famous. So they decided that they would perform some additional examination to check just to get to know more about him. And they found, for example, that there was 97% probability that Copernicus had blue eyes. And there is a painting of a famous Polish painter of the 19th century whose painting scenes important for the Polish nation. Yeah, they were like, because Poland was not present on the maps of Europe in the 19th century, so he was painting, we can call it patriotic paintings, yeah, just to support the nation, yeah, like scenes important for the nation, yeah. And one of his paintings shows Copernicus in his observatory, yeah, and he was painted with black eyes. So the press, the media, oh, the painter made a mistake because genetic studies show that Copernicus had blue eyes. Okay. Later, they also found that the Y chromosome of Nicolaus Copernicus belonged to haplogroup R1b. You remember, I told you today about the haplogroup R1b, which is specific to Western Europe, yeah? And you can see, okay, so these are this map shows the most frequent haplogroups, okay? So it doesn't, which is found in these different regions of Europe, okay? And you can see that haplogroup R1b, which was found in Copernicus, is found at highest frequency in Western Europe, okay? This region is labeled in pink, red. These are areas where haplogroup R1b is the most common haplogroup, okay? And I don't know if you know, but some people argue about nationality of Copernicus, whether he was Polish or German, okay? And now please take a look at this map. You can see that R1b is found at much higher frequency in Germany than in Poland, where haplogroup R1a is the most common haplogroup, okay? And so some people started claiming, oh, we have genetic evidence that Copernicus was German, not Polish, yeah? But I can tell you that R1b is also found in Poland. Approximately 12% of males in Poland belong to haplogroup R1b. So actually, it's not an evidence for ethnicity, of course, yeah? So just to tell you, what important things can be, what interesting things can be discovered when analyzing DNA of a person, okay? Here's an interesting story, a case of Lydia Fairchild. This is this woman shown on a photo with her 4 children. And this woman, I think, yeah, this woman had 2 children and was in the 3rd pregnancy when she separated Jamie Townsend, who was a father of her children. And she applied to a court for child support, yeah, from the father of the children, okay? And the court requested DNA evidence that Jamie was the father of the children, okay, just as it did in all such paternities, in all such cases, okay? And the results were quite surprising because results showed that Jamie was the results were shocking actually, because the results showed that Jamie was the father of the children, but Lydia was not the mother. And a lot of mess, what's going on, yeah? And the woman shows, come on, these are my dad. These are my children. Really, I gave birth to these children. No, what's going on? Yeah, she's totally shocked. What's going on? Yeah, there were even some suspicions that maybe there was surrogacy, or maybe she was even claiming benefits for other people's children. Yeah, that she said, "Oh, these are my children. I want money for these children." Yeah, there was a lot of different suspicions. Yeah. And she said, come on, these are my children. Really, I gave birth to these children. This is documentation from a hospital. And I said, how can we be sure that it was you who was taking birth in the hospital? DNA shows that you are not the mother. And I said, come on, I am the mother. I am in the 3rd pregnancy. I have another baby, and this is a baby, also the baby of Jamie. Yeah. And so finally the court requested a witness to be present during the 3rd birth, yeah, so the witness confirms, yeah, oh yes, I saw, yes, that Lydia gave birth to a 3rd child. So immediately after the birth, they tested DNA of this newborn, and again the same story. Jamie is the father of the newborn, and Lydia is not its mother. Okay, so now I have a question to you, a task for you. Please tell me 3 possible explanations, medical explanations, yeah. How can it happen that a woman regularly gives birth to children who who are shown in genetic examination that they are not her biological children. Okay.
So she was a chimera.
Chimera. What does it mean that she was a chimera?
Yeah, that she had at least 2 different genotypes, and the genotype from her ovaries is different than the rest of the body.
Yes, you are. I will write. Yes, you are right. Actually, she was a chimera, but how was she developed? How did she become a chimera?
So could it be that there were 2 zygotes in the body, in the womb of her mother, and they just fused together at the early stage?
Exactly. Yes, there was a fusion of, like, there were 2 embryos, like a dizygotic twin pregnancy, like 2 embryos or two zygotes. Yeah, probably embryos. We don't know at which stage exactly, and they fused. Yeah, they formed only 1 embryo. Okay, some cells from one of the sisters gave rise to the ovaries, and some cells from another sister gave rise to some other organs. Yeah, so this is how it was detected. Yeah, so this is the one of the explanations, which is actually correct. And I would like to find you some other explanations. When can it happen that a woman is shown in genetic analysis that she is not the mother of the child which she gives birth to? Cancer of the ovaries. Something. I don't know if cancer of the ovaries something dangerous and maybe the children. No.
Maybe something connected with organ transplantation.
Yes. What kind of transplantation do you mean? How can it happen? Yeah, just explain. Okay, let me check at the chat. Surrogacy, yes, surrogacy. Okay, so let's talk about the first hypothesis of Elizaveta. You told me that organ transplant, but what kind of transplant? How can it happen? So what happened in this case? What could have happened? What could have shown that she's not the mother?
Definitely her blood type could change.
But so what kind of organ would be transplanted? What do you mean? What could have been transplanted?
No, just a suggestion.
Transplantation of ovaries. I don't know if it was ever done, but it could. Yeah, but it would be a good idea. But something else, something which is more common. Bone marrow, yeah, something very common. Because because you told me for Victor, yeah, and Elizaveta, I will, yes, Elizaveta and Victor. Bone marrow transplant, because okay, you told me organ transplant. Okay, kidney transplant. Does it matter? No, come on, it doesn't matter. But bone marrow, if blood is collected, and nobody asked about bone marrow transplant, and the blood was collected from a mother and from the kids, okay, it shows that she's not the mother. Okay, so this is a good idea. And there is an idea of yes. So I'm asking when. Yes, the question was, when can it happen that the mother is shown not to be the mother of the children in DNA examination, even though she gave birth to the children? So a bone marrow recipient, a mother who is a bone marrow recipient and give birth to children will have, and we test the blood of the mother and the children. The test will show that she's not the mother. Okay. So this is another explanation, and another explanation which was provided by David is surrogacy. Actually, surrogacy is a situation that the woman gives birth to children for other couples. Actually, in this case, she was not, she was actually giving birth to children for herself, not for other people. Yeah. So surrogacy is not the good answer, but there is something correct in this question. Okay. So what would a mythical situation explaining that the woman gives another explanation of the situation, that the woman gives birth to children which are shown in DNA typing that they are not her children.
Could it be in vitro?
In vitro.
Like that there were two mothers. So, for example, the egg cell could be from the mother who gave birth.
Yes, exactly.
And
I agree.
the DNA material could be from the second woman.
Yes, exactly, Adrian, you are right. Another explanation and another medical explanation for such a result would be in vitro fertilization after egg cell donation. Yeah, so this is because not all in vitro fertilization will lead to such results, but what is important, it will be in vitro fertilization with egg cell donation. Yeah, that another woman donated egg cells to this woman, to this mother, and of course the sperms, the semen was from the husband, from the partner. Yeah, so this is how. So this is another medical explanation. Another medical situation which leads to such a result, which shows that she's not the mother. Okay, let's go further. Okay. Here is another example of application which concerns the third president of the United States of America, Thomas Jefferson, because there was a gossip that he had a son with his slave, with Sally Hemings. Okay, but it was many, many years ago. But geneticists decided that they would check it. What did they do? They found the male line descendants of two sons of Sally Hemings, of Thomas Woodson and of Aston Hemings. They found also mainline descendants of Phil Jefferson, who was a paternal uncle of the president, of Thomas Jefferson. And they found that the Y chromosome of Phil Jefferson, based on examination of his descendants, was identical as the Y chromosome of Aston Hemings, based on examination of his male descendants. Okay, so actually, this was moreover, this was a Y chromosome typical for European populations, and it's also very rare in European populations. So it was like a strong evidence that Aston was indeed a son of the president. Okay, so you can see it happened many years ago, but geneticists even proved that it was not only a gossip. Okay, another story from our casework concerned two men who came to us for kinship testing. Okay, the story was following. This is a case from our practice. Okay, one of them was a Pole, and another was an Italian. Another one was an Italian. Okay, so why did they come to us? We heard the following story: that during the Second World War in Italy, there was a Polish soldier. Actually, there were many Polish soldiers fighting in Italy then, yeah. And one of these Polish soldiers got to know an Italian woman there. They fall in love with each other. They even had a son, yeah. And when the man traveled back to Poland, he could not. He was stopped in Poland because he could not travel to Italy anymore because of the political situation in Europe. You know that Europe was divided into two political blocks after the Second World War: the Western Bloc, the NATO, and the Eastern Bloc, the Warsaw Pact, and he was moreover he was serving as a soldier in the West. So he was paid special attention from the communist government of Poland. Okay, so he could not travel back to Italy to his family, and the love story was over. And However, it was not over totally because, as I told you, there was this woman, Italian woman, gave birth to a boy. And when this boy grew up and was already a very mature man, he decided that he would find his father. And he was looking for his father very intensively. Finally, he found him, but it appeared that his putative father was already dead. But before he died, he established a family in Poland. He had a wife and several children. This Italian was not sure whether it was really his father. He didn't have any documents, yes. So he asked one of the sons of his putative father, yeah, who was dead, to take part together with him in genetic examination to check whether the father of this Polish man was also his father. Okay, so we tested two hypotheses. Yeah, you remember? I told you, remember the statistical examination, statistical analysis. Yes, so we always assumed two hypotheses. One of them was that there were half brothers sharing a common father. Yeah, so this was the first hypothesis, and the second one was that they were simply unrelated individuals. Okay, so we tested in total 19 autosomal markers. As many as 16 out of 19 markers showed common alleles between these two individuals. Okay, so I think it's quite many, 16 out of 19. Yeah, but what is, of course, it doesn't tell us anything about relationship because these alleles may be very common, very frequent in Europe. So of course we have to compute the likelihood ratio, which told us that 36 times, it was 36 times more likely that these two men were half brothers than that they were unrelated. Yeah, but 36 is not strong. Is not strong evidence. Okay, we need stronger evidence than 36 than 36. Okay, you remember that in Poland for paternity testing, to prove paternity, we need a likelihood ratio of at least 1 million. Yeah, of course, this is not a standard paternity testing case, it's something else, but 36 is not much. Okay, so my question to you is, what else could we do in this case in order to increase the evidence value? What else could we do? There are many possibilities.
Could we compare it with the background of Italian people, genetic background?
But you mean the autosomal markers?
Yeah, that like they are more like, because if those checked markers are rare in Italian population, it could mean that the chances of them being half brothers are higher.
Mm-hmm. Yes, I like your explanation somehow. Yeah, I like how you think a lot. I will even mark that I like your idea, but I can tell you that regarding autosomal markers and the alleles found in these two men, I can tell you that genetic studies show that European populations do not differ. Actually, for autosomal markers, we can use just like one population for the Caucasian populations of Europe. Yeah, so because they do not differ. Yeah, so this is something which will not help us, unfortunately. That's what would help us in this case to provide more evidence that they are half brothers, to increase the evidence value. Yes.
Could we check the Y chromosome?
Yes, exactly. Yeah, the Y chromosomes, because if they have had a common father, they must have identical Y chromosomes. Yeah. So this is actually what we did in our case. We checked the Y chromosomes because these were autosomal markers. Okay, so this is one of the solutions. We can check okay, Adrian, it was you. Yes, we can check the Y chromosomes. But so this is one of the solutions. What else could we do to increase the evidence value? We have tested 19 autosomal markers in these two men. Okay. Could we do something else to increase the evidence value? Hmm? The Y chromosome is okay. Mhm. What else could we do? We tested 19 autosomal loci, 19 autosomal markers. In these two individuals, I'm just trying to help you. We tested 19 autosomal markers in two individuals. Yeah, we can test more relatives for sure. The more relatives we have, the higher. So Victor, it was Victor. Yeah, the more. Yeah, this is a good idea. The more siblings we test, of course, the evidence value should increase. Yeah, there is not an answer on chat. Let me include mother's profile. Yeah, Martina, you are right. Martina. Okay. Yes, to include the mother, both mothers' profiles. Yeah, because if we know which alleles in these two men come from the mothers, then the other one must be paternal. Yeah, and if paternal alleles match, because right now we do not know which is maternal and which is paternal. Yeah, and if paternal alleles match identical, it will make the evidence value much stronger than in comparison to situation that we do not know the origin of the alleles. Okay, so this is a good way. We can include the mothers, we can include more siblings. So if we have the mother and if we have more siblings, then we can even reconstruct the profile, how the father looked like. Yeah, what was the profile of the father? Can we also determine the father's profile directly somehow?
From his grave.
Yes, exactly. Yeah, you are right. Okay. Yeah, we can simply exhume the body. Yeah, this is one of the options, which is the simplest for forensic genetics. Yeah, we can exhume the body and we have the profile determined directly. Another option is simply to test more STR markers. Yeah, more autosomal STR markers, because the more markers we test, the closer we will come to the truth. Okay. But we did not have DNA from the mothers, did not have DNA from other siblings. We did not have material from exhumed from the grave. Okay, we actually analyzed all the autosomal markers that we could at that time. So it was suggested by Adrian. Yeah, we analyzed the Y chromosomal profiles of these two men. And we found that the Y chromosomes were identical. Yeah, you can see they were identical. The likelihood ratio for the Y chromosome was even higher than for the autosomal markers. Yeah, even though less YSTR loci were tested. Yeah, because of the informativeness of the Y chromosome in this case. Yeah, because these two men must have identical Y chromosomes if they are related in paternal lineage. Okay, and the cumulative likelihood ratio was already almost 2000, so much better. Okay. And another question to you, which was already somehow answered, but not exactly. What else could we get to know about the Y chromosome of this Italian man to prove the story which I told you? I would like to get to know something more about the Y chromosome of the Italian man to prove the story which I told you.
Could we check the haplotype of the Y chromosome, because it could be different from the population of Poland?
Yes, it will be different from the population of Poland.
When the fathers is not the same. Like when they
no.
if they have the same father, the haplotype will be most likely RB2.
No. But please take a look. We already know that they have the same haplotypes. Yeah, can you see? We already know that both these two men have the same haplotypes. So they have actually identical haplotypes. But we would like to get to know something about the Y chromosome of the Italian. Let's imagine. Let's say that this is the Italian man. Yeah, the person number 2 is probably different. Okay, maybe I guess this is the person, the Italian. The person number 1, that the man number 1 is an Italian. And we would like to know something more about the Y chromosome of the Italian to prove the story which I told you. You are very nearby the correct answer. But what more can I get to know about the Y chromosome of the Italian man? Only the Italian man. I'm not interested in the Polish one. To prove the story which I told you. Actually, you told it. You told it, but I told you, oh, you are not right now. But the answer matches right now perfectly. This is what you told. Remember what you told at the beginning as the first answer?
Genetic background of the
Yes.
haplotype.
Haplogroup or haplotype? Genetic background of the haplogroup genetic background of the haplotype. But what for? How
in
can
Italian population?
Yes, we can compare this haplotype to the Italian population, and what? How can we interpret these results?
Like if this haplotype is not common in Italian population, that we can conclude this comes from the Poland.
Yes, exactly. Yeah, exactly. You are right. So, because according to the story told us by these people, this man was born in Italy, but his father was Polish. And you remember, you should know it already. I told you about the anthropology of the Y chromosomes, molecular anthropology of the Y chromosomes, that the Y chromosomes can be different even between closely related populations. And Poland and Italy, these are totally different worlds of the Y chromosomal gene pools. They are totally different populations. So there is an internet database which is used commonly in forensic genetics, which was I told you about this database, the Y chromosomal haplogroup, so the Y chromosomal STR haplotype reference database. Okay, and you can put your Y chromosomal profile, your Y chromosomal STR haplotype into this database, and you will see where it is found in the world. Okay, just look, when I did this examination, more or less the same number of Poles and Italians were tested. Okay, and the red dots indicate that this haplotype was found in this region. Okay, and you can see as many as 25 unrelated Poles shared this Y chromosome, shared the Y chromosome with the Italian who came to us for examination. Okay, but look at Italy, the whole country is blue. Not even one Italian man shared the same Y chromosomal profile with this Italian who came to us. So it was much more likely that his father was Polish than Italian. Okay, so we combined all these puzzles together and provided the final conclusion that these two men are half brothers. Okay, so I can tell you it was really nice to me to give them these results, because I can tell you it happens very rarely in my practice that everyone is happy about results of forensic genetic examination. Okay, because for example, this is this is forensic medicine. These are very often some sad stories. Okay, when I determine the identity of a cadaver, it is sad for the family. When I prove identity, when I when I confirm paternity, you can say, oh, the mother is happy. Yes, he will pay money to me. And the man will be, oh, I have to pay money to the child. Of course. But it's not always like this, because sometimes the man wants to be the father very, very much. It's not always like this. But generally, very often the woman wants the man to be the father. But in this case, I did for the first time something which made everyone happy. The Italian guy was happy because finally he found his father. He had a document that he is a son of this man. Yeah, he wanted. So we provided the final report also in English to this man, and with this document, he wanted to change his surname from Italian to a very difficult Polish surname. Yeah, I assume that his mother must have loved his Polish father a lot. Yeah, so this is why, and this is why he wanted to change his surname. Okay, and whereas the Polish guy was happy about these results because when I did this examination, Poland was not a member of the European Union yet. And it appeared suddenly that he had a family in the West, yeah, that he could travel, I don't know, for vacations maybe, but he could travel to his brother, that he had a brother in Italy. He could travel and visit his brother. It's nice to have a brother in Italy, isn't it? Okay, let's go further. I know if I told you about it already, but there is a cultural phenomenon which is common in many countries in the world, also in Poland, that that after the marriage, women change their surnames to her husband's surnames. Okay, this is common in many countries in the world, also in Poland. But in some countries, it's not like this. For example, I know that in Spain, women do not change the surnames after the marriage. Yeah, also I know in Saudi Arabia, because we have Arabic students, so they also do not change surnames. Women do not, but even if the women do not change surnames, children inherit surnames from fathers, not from mothers. Okay, paternal surnames are transferred. Okay, so if you combine this phenomenon with biology, you will see that surnames are transferred together with the Y chromosome, because Y chromosomes and surnames are inherited paternal. Okay, and you can use it for some genealogical investigations of males carrying the same surname. Okay, and we have an interesting case of a man, Mr. Andrzej, who is interested a lot in his genealogy, and he has a Polish noble surname. Okay, mine is a Polish peasant surname, and I know that there are not so many people carrying my surname in Poland, because there's a database, internet database, that you can put your surname in, and you will see where in Poland and your surname is found, and how often, how many people carry your surname. Okay, so I know that people carrying my surname there are not so many people like me with my surname, so I can assume that probably we come from one male ancestor, and we share also Y chromosomes. Okay, so but how it is in reality, I do not know. But this man has a Polish nobility surname, and he looks for men carrying the same surname as his. Yeah. And he wants us to compare the Y chromosomes. And usually he looks for some documents in some archives, in churches, to reconstruct the pedigrees, to find how the people are related. But sometimes he lacks paper documents, so he wants us to perform examination of the Y chromosomes. And he already sent us as many as 27 individuals. Yeah. Once there came to me even a big envelope from Russia. Yeah. My DHL company, you know, this courier company, DHL. I said, wow, to me from Russia, to my surname, yeah, to Russia by DHL. What is this? I did not order anything by internet from Russia. Yeah, I did it only once when I was traveling to Siberia. I bought a guidebook in a bookstore in, I think, what was the name of the city? I don't remember. Novosibirsk, I think. Oh, it doesn't matter. In a bookstore in Novosibirsk, but maybe I was saying, oh, maybe they sent me some advertisements or something like this. But why did they do it by DHL? Maybe they have a lot of money. Okay, I'm opening this envelope, which was to my surname, yeah, directly. I think maybe there was a poison, yeah. Russians are known for poisoning some political opponents, yeah. There was some poison. No, I opened this envelope, and inside there was a blood stain of a man who wanted to compare his Y chromosome with Mr. Andrzej's Y chromosome. Okay, just for me it was amazing how they found each other, because it was a time that there was, for example, the Facebook was not popular then. Yeah, so it was like, wow, how did he find this person, yeah, in Russia? Okay, let's go further. So what we found, for example, here you can see in red, we found we labeled in red alleles which are not found in, which are different from alleles found in Mr. Andrzej's Y haplotype. Okay, so and here you can see the this results presented graphically as genetic distances, between different Y chromosomal haplotypes. Okay, for example, this node number 1 is so large because there are several individuals hidden in it. Okay, they not only share the surnames, but they have also the same Y chromosomes. Yeah, the Y chromosomal haplotypes. Okay, person number 4 differed a bit, but you know that several hundreds of years there could have been a mutation. Yeah. Whereas haplotypes 10 and 13, they differed so much from person number 1 that you can say, oh, they were for sure different haplogroups. Yeah, I can say they were totally different haplogroups as well. Yeah, so I'm sure that they biologically they are totally unrelated. Yeah, paternal regarding, of course, the paternal lineage. Okay, so my question to you is, am I allowed to tell to person number 10, I'm sorry, you are totally that genealogically, historically, you come from another family. Okay, this is only an accident that you have the same surname as person number 1. Okay, that you are for sure you come from a different family. That historically you are not related. So, do you think I am allowed to tell this to person number 10? Very many. Look, married, like this is the distance from here, here. Large, huge distance. Many single step mutations or multi-step mutations. No, they are totally different.
We can't do it because we didn't check the DNA in the maternal side.
Yes, but the question of the man was, the question of the person number 10 was, am I related in paternal lineage to person number 1 because we have the same surnames? Am I is my family and is my paternal family and paternal family of person number 1 the same family because we have the same surnames? And I'm checking the Y chromosomes. I can see, wow, you have totally different Y chromosomes. So am I allowed to tell him, no, you come for sure from different families, paternal families?
Maybe there was an ancestor several thousands years ago.
Yes, you are right. Yes, you are right. Biologically, you are right, because all modern humans come from the same ancestor. Yeah, so paternal ancestor also. So of course, person number 1 and person number 10 had a common ancestor, probably tens of thousands years ago. But this is not the question of theirs, because they want to know about their relationship regarding common surname. Yeah, and the surnames were established, I don't know how many years ago, in different social groups at different times. There were like the first were like the city people who inhabited cities, then nobility and so on. So is it okay? I will make a different question. I will ask a different question. So I found a different how can I tell you? Okay, is it possible that I will have a son who will inherit from me my surname, my I don't know, my flat, I don't know, my nobility possessions. I don't know, I'm not from nobility, but let's imagine, I don't know, my emblem. My family didn't have any emblem, but is it possible that my son will inherit my emblem, my surname, my villages, whatever, if I were, of course, nobility representative, and my son will have a different Y chromosome from mine? Is it possible?
If your wife cheated, then yes.
If what? If what? Okay.
If your wife cheated, then you will.
Yes, you are right. Yes, if my wife cheated, this is one of the options, which is not socially, how to say, which is not accepted by this society to cheat. But there is a solution which is a situation which is socially accepted. What is it?
Could it be adoption?
Adoption, yes, exactly. Yeah, so actually remember that our genetic examination does not take into account such social events as adoption or cheating. Yeah, so this so just remember, yeah, that maybe historically they are related. Yeah, they come from the same paternal lineage. Yeah, and but something happened. I don't know. We do not know when, that the Y chromosome changed in the family, and we do not have. So remember that DNA does not tell us about such events. Okay, we are coming back for a moment to Yekaterinburg because according to the genealogical data, the Tsar's wife and all his children must have shared mitochondrial DNA with Prince Philip, Duke of Edinburgh. I think he died quite recently. Yeah, if I remember well, very recently he died. I don't know, this year or last year. I'm pretty sure it was, I don't know, this year or maybe last year. And he was a husband of the Queen of the United Kingdom. Okay, and he provided his DNA for comparative purposes, and geneticists found that his mitochondrial DNA was identical as the mitochondrial DNA of Tsar's family. And actually, so it was like a additional evidence supporting the royal origin of the remains, human remains that were tested. Okay, let us go further. Some examples of our reports. This is an example of DNA examination we performed in paternity testing. Okay, here is an example of an RFLP analysis. This is the enzyme which was used for digestion, the molecular probe, and in red, analysis was we have labeled in red analysis which is foreign, which is not found in the mother and the defendant. More foreign alleles, the conclusion that this man is not the father. Many alleles support, many markers support. Yeah, everything matches. Another story. Another case. Yeah, another case. Everything matches. We computed probability of paternity. This is the value. In the past, we needed only 5 nines. Right now, it is recommended to obtain at least 6 nines. And actually, we have proved that this man was the father of the child. Okay. Another case, a cadaver, burned body was found in Gdańsk. This is an old case which was analyzed by me in 2004. And his putative family was sent to us for collection of samples for comparative purposes. This was mother, brother, and sister. We collected the samples from the putative family members. We compared the autosomal markers. You can see amelogenin. Yeah, this is the male, and this is the mother. So this is the mother, the brother, and the sister. Yeah, putative mother, putative brother, and putative sister. We compared the profiles, and in all the markers tested, the men have the cadaver, the NN, had something in common with the mother, very often with the brother and sister, but of course, it's not obligatory. And I also compared the Y chromosomes of this NN and his putative brother, and actually, they were the Y chromosomal haplotypes were identical. And I also computed, probability that this man was a son of this woman was so high that it was a brother of this man was even higher, because the Y chromosome was included in the examination, and that he was a brother of this woman. There was only 81%, but it's normal for siblings. It's not always very high, because sometimes the alleles, so it's because the range of results can be really very, very different. Okay, so this was actually right now I could. So it was how we done it. It was how we did it in the past, because right now I would compute that he was simultaneously the son of this woman and brother of these two siblings. Okay, so this is how I will do it now. In the past, we were computing it separately for each relative. Yeah. So actually, I proved that this was this person. Moreover, just to tell you, I also computed the frequency of this profile in Poland, and in Poland is found once per 4. I don't know the English name, but in Polish and Spanish it will be quadrillion. 4 quadrillion people. Yeah, so you can see how rare. I think in English it will be septillion, I guess, but I'm not sure. So it's found once per. Generally 10 to the power of 24. Okay, so this is, you can see how rare this profile is in Poland. So if I ever detect identical DNA profile as in this man, I can be sure that this is this profile DNA from this person and no other person. Yeah, unless maybe this is a monozygotic twin or a clone. Yes, there are some other explanations. Okay. This is, I think, the last slide which I would like to tell you about. Yes, but I think we will keep this slide for the next meeting, because I would like to tell this slide as the summary, and we will have this case for solution. So next week we will have this exercise to do with the DNA profiles, which I would like you to solve. Then we will have this. Okay, and the analysis of biological states. Yes, so this is the topic which will be the leading topic for next week. Just the examination of biological states, criminalistic investigations, and something like this. Okay, so thank you very much. Have you got any questions right now? If not, see you next week.
Okay,
Bye-bye.
I think I would have a question, but not related to the topic of the class, more